Cool. Shall we make a start?
Sure.
Great. So, hi everyone. So for those of you who don't know me, I'm Elisabeth Kugler. I have done my PhD at the University of Sheffield and I'm now a postdoc at UCL. And so in our lab, we're interested into studying vascular development in the brain, and that's what we use zebrafish for. And so that's kind of why we're all here today, because we've developed this tool to actually quantify the zebrafish brain vascular architecture using Fiji. And so just before we kind of go into the science and the nitty-gritty and everything, I just thought I'd just let you briefly know. So the session will be recorded, so participants who weren't able to join will actually be able to watch this later after some editing, etc., etc. So they will get access to it. Secondly, we have at the bottom the Q&A function. So if there's any question, you can always just type in there, and I'll try to monitor the chat as well as the Slack as well as the emails. So in case someone drops out, has a bad internet connection or anything, just put it on the Slack if possible, or send me an email so we can kind of try and reconnect you, so no one kind of falls behind. Because most of us will be working from home, I am really aware that computers might not be great or internet connection or anything, and that's why I've asked you to download data. If you haven't done this, I will post the link in a minute into the chat, so please start it now, because for some people it actually took quite a while to download, again depending on the internet connection. But in this data file, I have processed all the data, so in case your computer is not strong enough or anything goes wrong, you have the individual processing steps on there, which we'll go through today. So I kind of really just want to make sure no one falls behind or you know, something goes wrong. So yeah, at the bottom you have the Q&A function, or chat function as it's called here, but also you have reactions. So we'll later have like introductions for everyone, and if you like something, it would be nice to just give a yay, you know, so we're not that disconnected from each other. And you should also be able to annotate in case you want to make any notes and stuff, but we'll go through that a little bit later. Are there any questions at the moment or anything unclear? Everyone happy? Cool.
Yes,
then I would share a video just as a brief like introduction for the background, etc. And while that is running, I will just make the admin and check the chats, etc. as well. Short presentation to introduce you to the background of the project, but also to give you a brief overview about the tutorial. So in our lab, we're using the zebrafish to study brain vascular development. And so for those of you who have never seen a zebrafish, this is what an adult zebrafish looks like. So this is a female and they're about 2 centimeters in size. However, we're using the embryos to really study vascular development and disease. And this is what the embryos look like. So this is a 4-day-old fish, and we look at the side views. So here would be the brain, the eye, the yolk, and this is the trunk. And so using zebrafish, what we can do is actually produce transgenic lines, like you can see here, to visualize tissues of interest with incredibly high specificity. And in this case here, it is actually the vasculature of this very same fish that we see here on the left. And so to visualize these data in our transgenic lines, we're using light sheet fluorescence microscopy, which is a technique where we have an uncoupling of the excitation and emission beam. And we actually excite a complete focal plane by using a light sheet and acquire this whole focal plane at once with this uncoupled emission objective. And so using light sheet microscopy, what we can do is actually penetrate quite deep into the tissue and therefore have a quite large depth of image acquisition, which is visualized here when we zoom in on the brain vasculature. So here would be the midbrain and the hindbrain would be sitting here. Here is the right eye and this is the left eye, so it's just rotating around. And so now it's kind of looking at the dorsal, so left eye and the right eye. And this whole section is about 700 microns wide and about 400 micrometers in depth. And so being able to visualize these data is really meaningful and very useful to us. However, this leads us to something that we call the data analysis bottleneck. So we have a lot of biology questions, experiments, and a lot of light sheet data. But now is really the question how we can bridge this gap from image acquisition to really having quantifiable results. And this is where data analysis sits. And this is really where my main work is in being embedded. And specifically in our lab, this means that we have a lot of data, especially in vascular development. So for example, in 2 and 3 days, we can see that the vasculature grows and the brain grows in size. However, it's unclear if, for example, vessel diameter, size, length, or branching points are changing. And this is even more challenging when we look at more subtle differences. For example, here where we have a control fish and one fish being treated with a anti-cancer drug. So this one is a vascular endothelial growth factor inhibitor, which is meant to reduce vascular growth. But just by looking at these images, it is really beyond any human perception to say if there is a difference or not. And so what we need is a computational workflow to really extract meaningful parameters as shown here, so branching points, volume, surface, radius, etc., to really describe the vasculature and see how the vasculature is being developed and what really its topology is. And so to do this, we have developed a workflow which is consistent of 4 main steps. The first one is image segmentation, the second one is registration, the third one is quantification, and the fourth one is biological application. If you're interested to learn a little bit more about them, you can also look at our bioRxiv paper, which has really a little bit more detail on them. But for today, I'll give you now an introduction about the individual steps and then what we're going to do today. So firstly, in terms of segmentation, this really encompasses how to understand your original data. So our data in 3D, but obviously we look here at them in 2D. And so what we need to understand is really how do the data look in 3 dimensions, what is their signal, what is their properties, is there any motion artifacts, and how can we really improve and enhance our data in a step called pre-processing, which improves the vascular signal and reduces the non-specific signal. By this pre-processing or improvement of our image quality, we can then perform image segmentation, which is a binarization process to extract the vessels. And this here is shown as a lookup table for depth, so the different colors show different depth, but it's really a binarization process where we set the vessels to 1 and the background to 0. Building on this segmentation, we then have image registration, which is a method to bring different embryos into one spatial coordinate system. So if we do the imaging, the fish look like this, but we actually want to be able to compare them. And so we try to overlap them, as we can see here, in 2D, and this is 3D. And by this overlapping and bringing them into one coordinate system, we can then actually study regions of similarity and variability as shown here. So regions of similarity are shown in yellow and regions of variability are shown in blue. Moving on from this, we then want to quantify these different vascular parameters, which I've mentioned before in the step of quantification, and then apply to different biological questions such as vascular development, disease, or treatment. And so to actually being able to share this workflow with the community, we have implemented everything into Fiji. And we also supply a graphical user interface, as you can see here, which has all the different steps of this workflow. And so users can just have a drop-down menu of choosing what they want to do. So you would just select this to yes or no. Sorry, my mouse is just jumping around. I just put it to the side. And so using this graphic user interface today, what we will do is actually we'll focus on 4 main parts of image processing. The first one is to understand our original data. So we'll look at them in 3D and assess how we can quantify image properties. We will look at image pre-processing using our graphic user interface, and we will also work on image segmentation as well as the quantification. And so we will kind of focus on these 4 points, but if you have questions on the other steps, please feel free to ask them either in the chat or later on. We will have plenty of time to really do a lot of discussion. And so to actually make it more interesting and really show you why this all this quantification is important, what we will do is apply to true biological data. And in this case, it is data where we look at how blood flow is impacting vascular patterning in the brain. And we can do this because fish can survive up to 7 days without blood flow. And so we can engineer them, or the DNA, in a way that they don't have heart contraction, and therefore they have never blood flow. And yet if we look at these fish, so this is a control, and this is one fish without blood flow, we see that even at 3 days, these fish without blood flow, or so-called TNNT2 morpholinos, have really changed vascular patterning. And so we will examine how this lack of blood flow does impact different vascular parameters. And at the end of the tutorial today, we will hopefully have all these different quantifications of vascular volume, surface, density, branching points, network length, and average radius. We will have two groups, so we will have an uninjected control and a TNNT2 morpholino, and we have, for the sake of time, not included the control morpholino, but this would be in an experimental setup, another group which we would examine here. And so with this, we'd hope to really kind of look a lot into this data analysis bottleneck and really examine the different steps of data analysis, including image understanding, enhancement, segmentation, quantification, and visualization. And so with this, I kind of now want to go into the tutorial bit and talk about the individual steps that we're going to do a little bit more. When we come to the individual steps, we will talk about them a little bit more in detail, but I thought I'd give you now a very brief overview about the individual steps that we're going to take today. And then, like I said, later, we're going to talk about them a bit more in detail. So firstly, like I said, we're going to look at data understanding because we work, or we look at our data in 2D, but we work actually in 3D with our data. And so we're using the 3D viewer to look at our data in 3D. And we will start to understand how different voxel properties actually impact your data, because when we look at our data, we think in pixels and voxels, and they are often assumed to be a cube, but actually often they are a rectangle, which means that in X and Y, if we look at our voxels, they might look like a cube, but actually in 3D, they are longer than they are in X and Y. And all this will make more sense when we actually look at it, but we will talk about why this actually matters. We will then also look at how to objectively assess image quality by a measure called contrast to noise ratio, which is a measure that if it's very high, it's easy to distinguish your signal from your background. If it's very low, it's very hard to distinguish visually as well as computationally what is a vessel and what is background. And we will do this quantitatively by using manual measurements. We will then look at how to improve and enhance our image qualities, and there are various different methods to do this, but today we're actually using a vascular specific method, which actually assumes that your vessels are a tube or a line, and so everything that is inside this tube or line or vessel will be enhanced, and everything that is outside will be being suppressed. And I don't want to go into the maths, but if you're interested, this is based on the Hessian matrix using SATA enhancement. And again, we will talk about this a little bit more at the actual step when we will do it. Going on from the enhancement, we will then do the segmentation, which is this binarization process. And in our case, what we're using is a histogram-based method. So if you look at your image, it has different intensity distributions. And if you look at the histogram, which is basically plotting the different intensity distributions and their frequency, as we can see here, what it will do is try to find two clusters, namely vascular and one background cluster, and then really distinguish all the voxels to be actually vascular or background. And this is what we see here. So this would be the input and this would be the output. And in this case, we use a threshold method called Otto thresholding. But again, there would be different methods to do this. Because also we look obviously at different fish, and fish can have different sizes, and we might image them at different angles, etc. What we want to do is actually extract the so-called regions of interest, or ROI or ROI. And we will do this manually to actually extract regions such as here, the eyes, or some background, or like some vessels which we're not really interested in. And this selection of this region of interest, when we apply to the different fish, it allows us then to actually have comparability between the different images. So if you'd imagine first you image the fish at different angles and maybe different positions, the selection of the region of interest will make these images more comparable. We then move on to actually look at the quantification. And in our case, what we do, the first step after having segmented and selected our region of interest, is actually to extract the vessel center lines. So here what you'd see is in red the vessels, and in yellow we actually look at the center lines. And the center lines are basically 1D or one voxelized representation of your data. And we will achieve this by skeletonization or center line extraction, with a method which is based on iterative thinning. So iterative thinning takes basically your object and it always peels out the outer layer until there's only one voxel left in the center, which is here shown in red. And this is what your skeleton is. And this skeletonization then allows us to extract the length and, for example, the branching points, as you can see here, because we can now extract the bifurcations or the branching points in our vessels. Moving on from that, we will use a so-called Euclidean distance map to look at local thickness or vessel diameters. And the way this works is that it uses the object, so the binarized object, and looks at the local thickness. And this is basically achieved by looking at how far different voxels are from the object border. So how far are they away from the background? And if we look at this example from MathWorks, what we see is that, for example, here in the horse, where we have a thinner leg, we will get a lower intensity, but the body is actually quite thick, and so we get a higher intensity. And so this intensity actually represents the local thickness or vessel diameter. And so when we apply this to our images, as you can see here, the higher intensity regions in yellow, they actually show us where the vessels are thicker, and the thinner ones are being the darker ones. And so we already have an approximation of how thick or thin the vessels are. But then to actually quantify it, we will combine this Euclidean distance map with the skeleton. And so we get a one voxelized representation of the individual vessel thicknesses. And this allows us to quantify the vessel diameter. And so with all this, like I said, we really hope to kind of engage all the different steps of data analysis with having this image understanding, enhancement, segmentation, quantification, and visualization. And so this was, again, just a brief overview, but we will talk about the individual steps a little bit more once we get to them in the tutorial. And just for kind of terms, or in terms of administration, we have now finished my kind of brief introduction, and we will now have a little bit of time for questions on all of this. And the next step will be to do the introductions from all of you and to look at them. And this is the list for the schedule of all of you. And I will also copy this into the chat so you know when it's kind of your turn to give the introduction. But with this, I also need to really acknowledge a lot of people who were involved in this project, especially my supervisors at the University of Sheffield, Paul Armitage and Tim Chico, who were really the main people involved in this project that I'm presenting here today. And so with this, I'm happy to take any questions. Right, so are there any questions so far?
I have a question.
Hi, Rupert.
Hi. Thank you for doing this. I'm really excited for the workshop. Is this segmentation applied to fixed tissue?
So in this case, it's applied to living samples, so the fish are alive, but you could fix the fish as well. However, if you do the fixation, what we sometimes find is actually that the vasculature collapses. And so we tend to normally look at living samples and how they develop over time.
And does it change, like, I know you said you have motion correction, but is your vasculature network changing in the time frame that you're capturing the image, or no, the time frame is too short, so it's a snapshot of the vasculature network in that time?
So the acquisition, so this stuff that I showed you, like the rotating thing, is a one time point acquisition, and that takes about 1 minute to acquire, and that's fine. So there is normally not really a lot of change due to growth. However, because the animals are alive, they can twitch and they can move. So we have looked into motion correction, and we're not talking about that today. But like I said, I try to keep a lot of like time for discussion, etc. So we can talk about this if someone of you is interested to look into like how we could address that. Because I am aware that especially if you do like imaging over a few minutes, there can be motion, especially with like heartbeat or fish twitching. And so, yeah, so for the short acquisitions, it's usually not that much of an issue. It's more an issue of longer time lapses. So if we look at time lapses over like 24 hours or something, then we definitely do need motion correction.
Thanks.
Awesome. Any other questions?
I have a question. So we'll probably get into the skeletonization process more later, I'm assuming, but I noticed just in looking at the skeleton that there were areas where it seemed like it was creating extra branches where maybe there weren't any. Is that real, or is that just from a layer that I'm not seeing in the projection?
So unfortunately, image analysis is never perfect. And so one of the things that we found the most challenging is actually extracting a reliable skeleton, because at the moment, what we do is using this thinning approach, which basically peels off the outer layers until there's only one left. And so what this produces, like you have observed, are so-called spurious branches. So you have branches where there wouldn't naturally be some. And so we can address this actually in the quantification of the skeleton. So in Fiji, you have this Analyze Skeleton plugin, which is what we actually will use. And there you can actually say you want to prune these branches. So pruning is basically taking these branches away computationally. And in the cases of Fiji, it's normally done based on length. So for example, if they are short, then they will be taken off, because most spurious branches will have just a certain length. They're just like leftovers. But you could also use different skeletonization approaches where you, for example, use the kind of the intensity, etc., and then you would get different skeletonization results. However, in Fiji, there's at the moment mainly this 3D skeletonization plugin, and this is unfortunately very prone to these short spurious branches for the vasculature.
Is there any way to actually correct it based on like the distance between branches rather than the length of branches?
What you could do, and what people have done, is actually more use the actual kind of proportion of like one long vessel to a short one, or for example, use the diameter. So some people have used how thick is the object in comparison to the length, and if they said it's actually thicker than it's long, then it's probably not an actual vessel. So people have addressed this, so you can kind of do it, but we haven't done it due to time. But yes, you could do things like that. All good? Cool. Right, if you're ready, I would say I kind of give the microphones basically to you. You should all be able to just, if you have a presentation, feel free to use it. If not, we kind of want to keep today informal, is really just kind of get to know each other. And also it's actually 4 people who I can't see, who are Amani, Rex, Atai, and Pascal. So I think we're 4 people short. So yeah, we're kind of on time with that. Right, and with that, Jean Young, do you want to go first and kind of just introduce yourself, who you are, what you do, why you're here?
Yeah, sure. Hi everyone. Yeah, I'm Qin Yun, and now I'm I'm a PhD student in Stony Brook University, in United States. I'm working in a cell biological lab now, and we are trying to do the cell segmentation and motif detection for the C. elegans cell data. And these data are fluorescence microscopy data. And we also need to do some like 3D reconstruction work in this recently. So I'm here to learn and to have more like communication about the how to quantification the data, 3D data, and also I want to know some like 3D reconstruction part work. Yeah. Thank you for coming here. Very happy to be here.
Great, thank you. Liz, you're next.
Yeah, so I am a postdoc in the Halloran lab at UW-Madison, and I think people might know me a little bit better from my work with the Huskin lab. So I study basically axon guidance mechanisms in zebrafish embryos, so during early development. And I use light sheet to do that. And it's actually very similar to what Elisabeth was introducing with her problem with the vasculature, where you have so we have this elaborate branched axon architecture that's going across the entire body of the zebrafish. And these branches are really hard to quantify by eye. You know, you can put 2 embryos next to each other, and it's really hard to tell those subtle differences. And so I've sort of gone the opposite direction of doing a very simple 2D quantification and automating that in order to very quickly, with our light sheet data, determine are there differences in the bulk population of axons, or are there not differences? But I would like to learn how to do a workflow for a more elaborate analysis and a more careful analysis, and that is why I am here, and I'm really excited to do that.
Great, thank you so much. And then I think Amani is not here, just double checking. No. Right, then Ripla.
Hi, I'm Ripla Arora. I'm a faculty at Michigan State University. I developed a methodology to image the mouse uterus from development all the way up until pregnancy. And we in our lab are developing tools, and we can stain for epithelium. So they have little glands that branch. we can stain for vasculature, we can stain for neurons, and we stain them and we have mutants that we know have problems. So first thing is recognizing by 3D these mutants that have been around for a really long time and they have pregnancy issues, but nobody has been able to look at them in 3D, and we're starting to do that. But whether we look at epithelial branches or blood vessels or neurons, we run into this problem of segmentation. So I've actually spent a lot of time with the Analyze Skeleton and Skeletonization plugin in ImageJ, and we also use an image analysis software that's not open source, which is IMARIS, because it gives really beautiful reconstructions. And that has a very specific plugin called Filaments, which is very useful for neuron tracing. So I've been trying to use all these different methods with the ultimate goal to segment and analyze branching patterns, whether it's blood vessels or neurons or epithelium. So I'm really interested to learn all these little quirks. And I think to me, it is also more interesting to connect with a whole group of people who are interested, so then we can troubleshoot and exchange ideas. And I think I'm really excited. I'm also excited to not do grant writing and manuscripts and administrative stuff and just spend like 4 hours looking at blood vessels. I'm quite excited. So thank you for doing this.
Great, thank you. It's great to have you with us, and especially not grant writing and not paper writing. I feel that. So yeah, agree. Great. Chiara.
Hello, everyone. My name is Chiara. I recently graduated with a master's degree in biomedical research at the University of Milan. I carried out my final master's project at the Department of Cellular Biology at the University of Valencia in Spain as part of an Erasmus program. So during this final project, we have developed a mouse model based on a single injection of stained fibrils, of alpha-synuclein preformed fibrils, directly in the bloodstream in order to study early and late effects of toxic aggregates in both the immune system and blood-brain barrier-associated cells. So we mainly focused on vascular alterations following the injection of fibrils. So we performed then histochemical analysis of CD31 marker, and we went we took some pictures, like some images with a confocal microscopy. So in order to quantify, like to do a quantitative assessment of vascular network, we went to see like some biological, really relevant biological parameters. So this one's length, branch length, intensity, vascular area, and density and thickness. And we're starting in doing this script, generating this script in Fiji. But we had some problem with the skeletonization and Analyze Skeleton plugin. So the thing that we were saying before, so I'm here actually because I want to know more about all these things. I want to also understand a little bit more about the light sheet microscopy, because it's kind of something I would be interested to learn more. And yeah, and that's it.
Great, thank you. Natasha.
Hi, everybody. I'm Natasha. I'm a postdoc in Boston in Sean Megason's lab right now. So I study the zebrafish blood-brain barrier, how it develops, and I come from a very genetics-heavy background rather than an imaging-heavy background. And so I've done some basic image analysis, but I am using zebrafish larvae, usually older ones, and I'm looking for the things that are coming outside of the blood vessels rather than staying within the blood vessels, but still learning how to really skeletonize and actually annotate the data and extract information in an unbiased fashion would be great. So that's what I'm trying to get out of this class today.
Thank you so much. Nagam.
Hello, I'm Nagam Youssef. I'm a second year PhD student at the University of Sheffield. I've just started my second year actually. So my project is about aging multimorbidity. So basically I'm trying to find the link between most chronic diseases that are caused by aging. So I focus on cardiovascular disease and osteoarthritis. And I try to find the common genes between them using GWAS data. And then I try to do some manipulation to these genes using CRISPR-Cas9 or some inhibitors in zebrafish embryos and see their effect and hopefully try to connect downstream pathways as well. So I do all this in zebrafish embryos. And that's why I use the light sheet and other microscopes as well. They're very advanced and really complicated. So I'm here to learn more about imaging the cardiovascular system in general, because we're an osteoarthritis-based lab, so we're really good at this, but we're not that good with the cardiovascular work. And I'm also trying to see if I can possibly apply the same concepts to imaging the musculoskeletal system. Yeah, so that's it.
Thank you, Nagam. Mariana.
Oh, hello everyone. I'm Mariana. I actually just started my PhD 1 month ago, so very fresh. I did my master thesis in zebrafish and how the vascular response occurs after spinal cord injury. I used adult zebrafish, and that's a project that I'm continuing in my PhD project, and also combining the vascular response with what kind of cells might be influencing this response and modulating vessel remodeling, for example. And during my master thesis, I tried to use a 3D approach to quantify these things actually, but it didn't work. So I had to take a more 2D approach. And so when I saw this workshop, I got really excited. And of course, I signed in immediately, and I'm very happy to be here.
Great, thank you so much. Just need to check the list. So Rex, I think, is not here. Octavio.
Hi, can you hear me?
Oh, yes, I just muted myself, yeah.
Oh, I think I'm a older guy here. I'm a Dr. Octavio Mercado. I'm from Mexico City. And my work is about the morphological changes of astrocytes and microglia. So I think this tutorial will be useful for me because I would like to measure in 3D these morphological changes after status epilepticus. I'm working with status epilepticus and epilepsy.
Thank you so much. I think we have lost Sayat. Yeah, it's gone. Okay. Alba.
Yeah, hi everyone. I am Alper. I am a faculty member at Koç University in Turkey in physics and electrical engineering. So I work on the instrumentation side. So I built confocal microscope, structured animation microscope. We have built an open spin-based light sheet microscope. I also work on microfluidics. So I am collaborating with neuroscientists for brain vascularization data analysis too. So we have data recorded with a multiphoton microscope, vascularization data, and we are trying to do 3D visualization of it. In fact, we are also working with computer scientists and see if we can also develop some solutions, not on Java based only, but maybe other like platforms, Python, etc. But I am not the expert. I am the instrumentation guy. In fact, Atay, who is not here, is my colleague on the neuroscience side. Thanks.
Great, thank you so much. And I think Atay isn't here and Pascal isn't here. So Andre, last but not least. Please.
Hi everyone, do you hear me? Yes, okay. I'm from Portugal, from the University of the Algarve, and I am a first-year PhD student. I started my PhD work more or less 1 year ago on the group of Dr. Marco Campinho that focuses on the role of the thyroid hormones in vertebrate development. In this project, I am developing CRISPR-Cas9 based loss of function mutant lines of each receptor in zebrafish. And at this moment, we are already crossing the F1 generation fish and trying to identify the phenotype for each mutation. With these mutant lines, we want to functionally dissect the genetics dependent on the maternal thyroid hormones that are triggered by each receptor. I will be using technologies such as immunostaining and in situ hybridization. And for image acquisition, I'm already using light sheet. We know that the thyroid hormone is absence profoundly modify the later dorsal specification of the brain and the spinal cord, as well as the vasculature is compromised. So I will need to quantify the image, and this course will be very useful for me, I think. I'm very happy to be here. Thanks for doing it.
Great, thank you so much. So I think that really shows that we're all like a bunch of like different fields and different stages and different parts of the world, which is really great. And so thanks again for joining. Right, does anyone have questions, burning, I don't know, thoughts for anything, or are we all good to go to the next step? Cool. Right, so then with that we can actually start and go into the hands-on practical. Right, you should actually be able to see my screen now. And I'll just open this here as well, just to check. Right, I'll just close all of that. Right, so first things first, I've sent you all the PDF file, which has basically all the individual steps in it, but we'll go through all of them today. Obviously make notes for things which are unclear. If there are any questions at any time, feel free again to drop them in the chat or just interrupt me if you get lost with anything. I just want everyone to really learn something today and not just kind of load a lot of knowledge on you, but really have kind of discussions of like what is useful and what you gain out of everything. And so in the PDF file, first thing on page, I think 3, yes, page 3 is actually how we kind of get started. So I hope all of you have downloaded the data. If yes, please hopefully have downloaded them, as well as have decompressed them to basically extract the zip file. And I'll talk you through what is in the folder in a moment. But first, I hope that all of you also have Fiji on your computer. And what we do as the next step is actually update it. If you have never updated your Fiji, if you want to open your Fiji, on the right side, you will have a help button. And then a button it says update. And then it will run for a little bit. And then you can say show details. And on the left, and then you can open like this window where it says manage update site. If you get to this point, you can then click neuroanatomy and close. So it sounds more complicated than it is, but I'll give you all just a few minutes to kind of just open your Fiji and then go to this help update, run an update, and hopefully add this neuroanatomy to your Fiji. And then you just need to restart your Fiji distribution just so it implements basically this update. And the reason why we do this is actually because later when we do skeleton analysis, this neuroanatomy plugin actually allows you to summarize your skeleton. So it gives you a very nice quick number of junctions, etc. Because normally in your analyzed skeleton, you get a lot of like sub branches, but we don't want that. We want to have one number for the complete image. If anyone struggles, raise your hand or just unmute yourself or ask a question. If everything is all right, you can just, or if you have done it, you can just drop a yes into the chat so we all know where we are. Yes, Nagan, so it's updating everything. Yes, that's all right. The main thing that you want to get is basically this page where you can say manage update sites, and then you click this neuroanatomy in the list.
Hi, Elizabeth. It is in my list. Do I need to do update? Also, I mean, in the updater, it says updated, updated. Do I need to do a separate update, or it's enough that it's in the list?
It has to have the neuroanatomy in the list. So if you have ticked that before already, then that's fine. But most people won't have this in the list. That's why we basically just do it. But if you have it already, then it should be fine.
Okay.
Okay, that should be almost all of us. Just, okay. Did everyone manage now? Is that all right? I think Andre's one of the people who hasn't said yes yet, and Alpa. Brilliant. Perfect. I think and hope that's everyone. Right. So once we have done this, which is like I said, for summarizing the skeleton, if you haven't done it, what we also need to download is actually the macro that we're going to use. And I'm trying to just copy that into the chat as well. But that should only take a few minutes. If I can open the chat, because that's in the list. So that's the link. So you can download that on GitHub. So this is the actual code that we're going to show you. Looks more complicated than it is. All that you need is this sdoc2 and click on this to download the macros, and then it downloads the zip file, and then you can actually decompress it. So these are the macros that we're going to use today. And then if you could give, again, like a quick yes, once you found it or once you downloaded it, that would be great. So we know we're all again on the same stuff. Okay, that's again almost all of us. Again, if anyone has issues, just wave, let us know. But that's the main steps really. Once you have extracted it, we can either drag and drop it now into the feed, you can do it later. But I hope all of you have now your distribution open in Fiji. And now what we kind of start going into is actually really starting to understand and work with data. So if you look at your data file, which we hopefully have downloaded and extracted, you will have your data where it says I2K data 512 by 512. And the reason why this is actually that we have downsampled data. So normally they would be far larger. They're almost like 4 times the size of what they are, just to kind of be able to be run on a normal computer. And so this is why we downsampled them to 512 to 512, because I thought most of us are normal human beings with a normal computer, which does not work with light sheet data. So like I said, we have 2 different kind of groups in here. The first one are uninjected controls, which are healthy fish, which are 3 days old. So 3 DPF means they're 3 days, and uninjected control means they have nothing done to them. E4, E5, E6 is basically just embryo 4, 5, and 6. And then dorsal means it is acquired from the dorsal position. So we could obviously image the fish from different directions, but we have done it from the dorsal, so we look at the back of the head from the fish, basically. So these are these 3 examples which we have here. And then we have 3T and NT2 fish, which are these fish without blood flow. So they have never had any blood flow and therefore have obviously a changed vasculature. In this, you also have the maximum intensity projection. So if you look at this maximum intensity projection, and actually adjust the contrast a little bit. I'm just trying to move this. You would see that these fish actually have quite normal vascular patterning. So this is what we would expect. So here again sits the midbrain, the left eye, the right eye, and here sits basically the dorsal vasculature. And if we compare this to one of the fish which has no blood flow, what we see is that they have actually a smaller brain, so it's kind of a non-inflated brain. And here where the midbrain would be, we actually see this major change in vascular architecture. So again, this is control, and this is our fish without blood flow. And so this is kind of one very, very crude example, but normally, obviously, we would look at more subtle differences. And the more subtle differences is really where the quantification would make sense, because like I said in the presentation before, if you have like an anti-cancer drug, for example, you really want to know if there is a difference or not. But for the sake of today, we use these examples where we have really a major difference. So we all kind of see what we'll get out of the data. Okay. Can I just have a quick hand up or yes, if everyone is at this stage and has the data and has everything there, so all good to go. Yes, Liz, you're raising your hand virtually.
Sorry, I just meant I heard hands up. And like, yep, okay. Got it.
Okay, cool. It's a bit complicated because I'm like on my main computer and then I have the other one to monitor things. So I'm always a bit confused here. Cool. Right. Once we are at the stage, what we'll do next is actually look at the data in 3D, like I've said. And so to do this, you can just take one of these uninjected controls and just drag and drop them into Fiji. So these are normal TIFF files, so they are they should be standard for each Fiji distribution. If we zoom through, what we do, because it's a 3D stack, just for those of you who have not really worked with 3D data or Fiji, I'll just make this smaller here. It's basically a 3D stack of data, and so what you see here is basically the slice number where you are, and this is the total slice number, and then it gives you the microns in size as well as the voxels, and this is what I've mentioned before. We have downsampled them to 512 by 512, but normally they would be like almost 2000 by 2000. It's a 16-bit image, and this is the size of the actual image. And again, for those of you who would normally work with light sheet data, they know that a normal light sheet image is more like in the 3-5 gigabyte range. So this is actually a very small image we look at. If you go through and just scroll through, what you'll see is that this is actually what the brain vasculature looks like as a stack. And so at the bottom, so the first slide is the ventral part. And once we go through and basically go to the right, we'll actually go to the dorsal part. So about here is in the middle. So this is the midbrain section. Here would be again the hindbrain. And if we go to the back, these are the vessels, which are the very dorsal part, which basically cover the midbrain of the fish. So if you all just want to drag and drop one uninjected controls, and then just let me know if your computer can cope, or if we already have some computational issues, that just would be great. So just if you have issues, just let me know at any point in the chat. Great. So far everyone seems to be, or every computer seems to be fine. Right. So what we want to do next is actually to look at the data in 3D, because like I said, we obviously work with 3D data, and this is a step that I find a lot of people kind of dismiss, and therefore a lot of issues actually creep in. And so what we'll do is actually just go to plugins, and then you should have the 3D viewer as a plugin on there. If you do this, it might throw up an error message. I normally just ignore that. And then it brings you this console up where it basically asks you what to do. It should be fine to leave all the default settings. You could change it to show the surface. You can have a different color, etc., etc. But for this case, we can leave it. If you see that your computer is actually struggling, you can resample them even down a little bit further. So normally they are resampled by a factor of 2, which basically means it decreases image quality, but increases computational speed. So if you realize, okay, your computer really struggles, just resample to a factor of 4. If you see your computer is really fine, you can resample to a factor of 1. But that obviously means with 1, it just will take longer. If you do that, it will ask you to convert it, and you can just say yes. And then it will take a little bit to actually just 3D render the data. And this is what we get. So now you should have this window open, and you can actually click and just rotate. And with this, you can now actually start to investigate how your data look like in 3D. And I find this very helpful because often we kind of forget the data are 3-dimensional, and therefore a lot of kind of issues creep in, and we'll talk about this in a minute. But I find it kind of really good to see rotate, just, okay, where are we, get an orientation again. So this is left eye, this is the right eye, forebrain, midbrain, and hindbrain. And then you can rotate that around, and you would be actually able to look at the embryo basically from the bottom. So this is the ventral part, and this is the basilar artery, which is the main vessel, filling the blood into the brain.
This is a really naive question, but so
go ahead, that's what we're here for.
Yeah, so while I'm like moving it around, is there an easy way to actually move the whole 3D structure over?
As far as I know, not really. If someone knows, great. But I personally, I find just this rotation is normally kind of the easiest. If this kind of thing comes up, I find it really difficult because everything starts moving around. So I'm personally not a fan of that. But you can zoom in and can zoom out with controls. But to physically move it, like to the left and right, I wouldn't know if that was possible.
Okay.
If that answers the question.
So I know that if you have a mouse that has a scroll button, if you press down on the scroll, then you can move the image in your screen. If you just scroll it up and down, that's zooming in and zooming out. But if you press, so you need the mouse with the scrolling.
Okay.
Yeah,
I'm trying to do that right now. How do I do that? I can't do it.
If you press it, then you can move it around. Well, yeah, I'm able to do it.
Do you use a Mac or Windows?
Mac.
I use a Windows.
It might be a Mac thing because I do Windows and I can't do it either.
I tried to do it. Well, I don't have a mouse right now, so but in Mac, it's also not doable without a mouse.
Okay. Interesting. Never knew that. Cool. Great. If we're all happy with that, we actually go to kind of really back to the basics, namely looking at what the voxels look like. Like I said in the presentation, which I'll just quickly reopen here so we know which slide I'm actually talking about. What I briefly talked about is actually voxel properties. So we acquire our data in 3D. And so what we look at when we look at a stack is normally the X and Y direction. Again, this is the Y and this is the X in an image. And the different planes are basically the Z, so the depth that we go into the image. And so because we normally look kind of from the top, we always kind of assume that our vessel is like a very nice cube, but it's normally not. It is like a rectangle, so it's elongated. And this is what's called an isotropy of your data. And this is most of the time not really an issue because people don't consider it too much, but for those of you who do work with image analysis, you will know it can be really an issue, especially if this Z is very long, because obviously it tries to interpolate the data. So if you, for example, acquire an image and then you have like 10 micron steps, it just can't interpolate the data. And so everything will be stretched significantly. And so to look at this and kind of your voxel properties, you can go actually to image and properties, and then it will tell you what your width, your height, and your depth of the voxels is. So in this case, it's obviously changed because I did this resampling, but normally we'd have 0.3 by 0.3 by 0.5-ish. So we would have a longer voxel than actually the width or the height of the voxel. And the thing why this kind of is really important is also because there are processing steps which actually accidentally change your number of slices with your frames. And then you don't know what happened, but this is one thing to look at. So be very careful with this. And also there are processing steps which will actually artificially change your width, height, depth to 1. And then suddenly your data look completely squished and you kind of wonder what happened. And so this is one of the really kind of basic things where you waste hours just because something simple as that happened. And if you kind of just want to have a play with how that would look like, you can actually change these. And then look at this again with the 3D viewer and load everything in completely the same. And then what you see is actually, in this case, it significantly just stretched the data. But the typical thing is that your data suddenly are massively squashed or in like really weird in one direction. So this is kind of the simple things. But if you'd look at the stack itself, you would never know because it looks exactly the same to you. But again, this is 2D and this is 3D. So this is one of the, this is where you lose a lot of time when doing image analysis. One very simple thing like this, looking at your image properties. Okay.
Elisabeth, how did we enter this Z dimension information?
If you click on your TIFF file,
okay,
and then go to image properties,
okay,
then it should open up this little window where it shows you the channels, the slices, and these ones.
Yeah, thank you.
Welcome. And it's same as also it can like sometimes change your microns to pixels. So these are kind of the really, like I said, it sounds so simple, but this is where you lose most of your time just finding this, like, why does it not work? Right. Is everyone happy so far? Yeah. Cool. I just close this TIFF again because we have changed the properties with it. So I just close all of this and just drag and drop it in again. And then we want to look at contrast to noise ratio. So I just go to the presentation again. Contrast to noise ratio is basically this measurement where we want to look at how good or bad your data quality is. Because it's very easy to look at your data and say, oh, it looks good. But especially if you do some processing, you need to have a quantitative assessment of, did it really change something or not? And so in our case, what we found is that this contrast to noise ratio measurement was really kind of a good step to look at the quality of the data and how this changes during image pre-processing. And so, like I said, it's a quantitative measure of if it's very high, it means there's a good quality. Anyone else having issues hearing me? Ripple, am I breaking off or am I just too calm or anything? Do you want to turn off your video and see if that makes it better? Maybe it's the bandwidth.
Should we all turn off our video? That might help.
Well, I can do. I don't. Yeah. Where's my window? Okay, Ripley, can you just let us know if that works better or? Thanks, Liz. Okay, I'm not sure if Ripley actually logged out or she's still here. If okay, she will hopefully let us know. All right. Okay, I'm just continuing. Okay, now she can hear us. That's good. Great. If Octavia, Ripley, if you just want to also turn off your video, if that really makes the quality better, that would be helpful. Thank you. All right, so contrast to noise ratio is basically a measurement where we're now going to measure the mean signal of a vessel and subtract it by the mean non-vascular signal, which is still a region of interest in the brain. And the reason why we look at this non-vascular region is actually that if we look at living tissue, there's often really a lot of autofluorescence and background, and we need to consider this. And there are certain tissues which have more autofluorescence, certain tissues which have less autofluorescence. But taking this measurement as well really gives us kind of a comparison to what is in the tissue, but where we have no vasculature, if that makes sense. And then we divided by the standard deviation of the background, which is basically a region of interest placed outside the fish, somewhere in this case in the embedding medium, which is agarose, but it could be anything literally. So to do this, this is a manual process. And what you actually do is you look at your vessels and you find something that you can actually compare. And the good thing is in zebrafish, they're actually quite comparable between fish. And what we look at is normally this basilar artery, which is at the bottom of the fish, and say this is more or less in all the fish there. So we can use this vessel as a standard to measure the contrast to noise ratio within them. So what we're now going to do is actually open the region of interest manager, and we do this by edit, selection, and then add to manager. And then what you get is the so-called region of interest manager. If we then go to the circular ROI, or region of interest, you can draw like regions like this inside the vessel, or you can look at a different one, whatever you want to do. But in our case, like we said, we kind of choose these regions of interest. And then to actually measure the intensity, we can go to analyze and histogram. And what this will give you is basically the intensity distribution. So a histogram gives you the different intensities in your image. And in this case, we don't want to look at the stack, but we just look at the specific vessel. And so we just say no, because we just want to look at it in this individual plane, just for this example now. What this will give you is basically the intensity distribution. So this is the frequency, and this is the different intensities. What you can also do is actually change this first from a 16-bit to 8-bit. So it basically limits the intensity from 0 to 255, and then you can compare it between fish a little better. And then you get a mean signal as well as a standard deviation, loads of other measurements. But in this case, we're actually interested into the mean of the signal. If you'd want
that, sorry, Elizabeth.
No, go ahead.
How did you change that to the 256, or you didn't?
So I haven't in this case, but what you would do is go to image, type, and 8-bit.
Gotcha. Okay.
Thanks. Right, and then Histogram. There we are. And so now it's limited. And it should have done with you. One really handy trick as well is so you could add this region of interest now to your ROI manager. So this will save it. So if you move around like somewhere, like hit this and then just click here, it will go back to this. And so if you do some processing, you can then basically save this by going to more and then save. You could say this is my embryo 1. Vascular 1, for example, and then it saves this region of interest. And then if you go to your histogram and actually click on live, what you can do is actually either drag and drop this somewhere else, and it will actually change your values. So you can do it like this, or you can just draw a new region of interest. And so, for example, outside the fish, this is where we'd look at the standard deviation of the background to do the measurements. I'd like you to all kind of have a try and just do these measurements, but I actually did draw the regions of interest for you, just to save you time if you want to. And so in your folder, I2K data, you have something that's called a ROI set, and then one which is CNR ROIs. And the CNRs here, these are the ones, so one set for vessels. So if you drag and drop this, it will open it for your uninjected fish and your fish without blood flow. Same for non-vascular, so these are these 6, and then the background ones are for the standard deviation. Okay. Is everyone clear so far, or are there any questions or issues? Brilliant. No questions. Great. Thanks.
So did you add those ROIs from a file, right? All
yes.
those.
So I drew them for you guys. So you don't have
that, I think.
Pardon?
I missed that point. How did you like put all those ROIs to the ROI manager?
So I drew them for you and I've saved them in CNR. So it's in the I2K folder,
okay.
CNR ROIs, and then you have these 3 sets. So the first one is for the vessels, one is for non-vascular, and the third one is for the background. And basically all that I've done is I went through all the images and placed these regions of interest and then said add and then save them as whatever name you see here. And then what you can do is actually you can, for example, select multiple of them and then go more and save, and then it will save it as a ROI set for you. So I find that very handy to do that for my experiments.
It won't open. I'm trying to drag and drop, and it won't open the ROI manager.
You can try edit selection and then add to manager. So open the manager first and then drag and drop it. Maybe that.
And are you dropping it in the manager or in Fiji?
Into Fiji.
Yeah, it sees drag and drop, but it's not adding them. I don't know why.
Okay. If you go to more and open. Which file? If you go to more and open, you should also be able to just select it and then open it, and then it should just attach it at the bottom here.
Which open option did you use? Where is the open option?
So in the ROI manager,
okay.
Yeah, if you go to more.
In more, okay.
And then open. It's a bit hidden, so yeah.
Okay, clear.
Anyone having issues now, or does that work? Okay, I assume that this works.
Do we really need to save the ROI or not?
So the reason why we do save it is actually it will make sense later when we look at the enhanced images. If you just look at your original data, you won't see that much of a difference. But what we want to look at is actually what happens when we process the data, especially with our enhancement.
Okay.
And so that's kind of why I've actually saved it for you, because it is quite laborious. I mean, you get used to it, but it's one of those things where you kind of have to sit down and do it and save it. But that's kind of why I drew it for you. Basically, what we want to do now is for these 6 images, if you just say, for example, you drag in your first one, and then you look at which fish we're looking at. So for example, this is the uninjected one, and then go to your vessel, and then it jumps to your vessel straight away. And then if you go to your histogram, it will actually, oh, sorry, we need the 8-bit conversion first, sorry. Type an 8-bit, and then go to your, analyze histogram. And then you will get the mean values, etc. And then you can write them down for your different fish. And then basically, so what we want to do is we want to have one file where we look at the contrast to noise ratios like this. So you have the mean vascular signal for these 3 fish and for your 3 treated fish, and then your mean non-vascular signal and your standard deviation of the background. And then you just can fill it in like that.
Okay, maybe I missed this, but how are we supposed to import the CNR ROIs that you already annotated?
So either you can go to, so you can either drag and drop the folder. So it's in your data folder in the I2K 512 in the CNR ROIs. So you should be able to drag and drop. Or if there's an issue, you can go to your manager, more, and open.
Okay, so I have to unzip the CNR's ROIs first. and
No,
then-
it should. That's the thing. So these sets, they are always saved as a zip, but they will be decompressed automatically by Fiji. So you
Okay.
should be able to really just
Mine's not
open it.
letting me open either the uncompressed or the compressed.
How have you tried it?
To try drag and drop it.
Okay, can you go to your edit selection and add manager? So just open the manager like that.
The add to manager or
Yeah.
I had to open each of them individually, and then it let me open all of them. But I had to open them all individually.
Really?
It may just be something with Fiji, or I don't know. But
Are you both using Mac though?
yeah, I'm using Mac.
Okay, so that might actually be an issue then. Okay, I never knew that. So ROI.
Yeah, mine's not even like opening anything. Okay, so then I have my ROI manager and then
If you click on more, through more, you go to open, and then it won't let me select multiple, but if I click on one at a time, it'll let me open them.
Gotcha.
Okay. Well, we don't have to do all of them. It's more like a proof of principle thing. You can also just do like one fish, two fish. The main thing that we want to look at with this is how we improve image quality with the enhancement. So if you just want to do it for like uninjected fish 4, let's say, that's all right. It's really kind of, you don't have to do all of them.
It was okay actually if I did it through the more and then did the zip. But
Okay.
yeah, we cannot drag and drop with the Mac.
That's good to know. I'll make a note of that. Thank you.
I could open a total of 18 ROIs. I see more than 18 ROIs in your screen.
Yeah, because I just opened some of them multiple times just to show people. Thanks. But no worries, thank you. Right, and if we have all of that, like I said, if you just want to note down the mean signal, so if you do fast 8-bit conversion, so click on open your image, click on the image type 8-bit. It's an 8-bit, and then yeah.
Yeah, why are we doing the 8-bit conversion? I mean, for convenience,
to make it just more comparable between the fish, because in this case, if we limit it to the 255, we just make it more comparable, but you don't have to, because in this case, we will expect to have a very significant difference between the unprocessed and processed ones. And yeah, so like I said, if you then click on your vessel, so for example, here we have Uninjected Control Embryo 4 vessel, and then go to Analyze Histogram, and then note, you get your mean value, so you can write this down. And if you then click live, go to the side and click the image again, you can actually go like this, so you can jump around like that, and it automatically changes it to full. And then if you go to the background, the standard deviation is automatically changed to this 0.06. So this is just one example. You can do this for one fish, or you can do this for 2, 3 fish. It's just to show you kind of how it works and how you could do it. Okay. Should I just give you a few minutes doing this? And then you just let me know when you're kind of happy, or if you have any questions.
Are we still just selecting, like doing the measurements of the CNR? Sorry, I stepped away.
Yeah, no, yeah, like I said, if you want to do it for one or multiple ones, you can do it. It's just really for everyone to kind of get a feeling about how it works and how to kind of work with these ROIs. If you want, you can do all of them, or you can do that at a later stage. It's really kind of just to give you a feeling of how it works.
Okay, thanks.
You're welcome. And yeah, once you had all kind of enough of it, just drag like or drop a yes into the are enough into the chat so we know we can move on from this.
Sorry, I have a question.
Yes, please.
Like once we have like the mean signal value for the vascular, like the vessel, and for the background and for not the vessel, like once we see from a single condition all those means value from the histogram, this enables us to calculate the CNR or not.
Yes, so this allows us to quantify the CNR. So basically, if you would have a complete data file, where you have, so you basically start to collate an Excel file like this, where you have all your values. Let's say you have 26 in your mean vascular, you have 6 in your or 5 in your non-vascular, and like 0.0, let's say 5 in your standard deviation. The formula is basically your mean vascular minus the non-vascular, and then divided by the standard deviation of the background. And what you get is a value like this, and in this case it's 420, which is actually really, really high. So in fluorescence microscopy, we have very high contrast to noise ratio measurements. For anyone who works with MRI or CT, they work with CNRs of like 3 or 5, where it's like, that's the so-called Rose criterion. So everything that's above, I think, 5 is really, really high. So 420 is incredibly high. However, we actually, when we do our pre-processing, we will actually increase this even higher. And so we can improve the quality even a little bit more. And so you have a quantitative measure of what your data look like.
Okay, yeah. Thank you.
Did everyone get that? I hope that was makes sense. Brilliant, thank you. It's great to have a bit of feedback in the chat because it's very strange just to talk to the computer.
Do we need to make the Excel? Is that of use for us right now, or that was just an exercise to know how to calculate?
You can do. I mean, I would always recommend it, but if you, I think for the sake of like discussion and stuff, you don't have to do it for all of them. It's just like I just very briefly did something like this, so that you also have the formula and then just save it. It might be useful for some of you for later on, like in your own research.
Got it. Thanks.
Elisabeth, how necessary is it to always have the same like blood vessel?
It depends. So what we found is actually in fish, it's the contrast to noise ratio is independent of the vessel that we look at if we use this specific transgenic. But obviously, if you have a specific gene, etc., that might be differently expressed in different vessels. Or if you have tissues and you look at one vessel which is very superficial and the other one is really deep, some staining might not get through, or the light penetration in the microscope doesn't go through that far. So you might have differences based on the location or like the anatomy or the staining, but also you can have differences based on the size of the vessel, because obviously the larger the vessel, the more membrane. And if you have, for example, membrane staining, that could obviously impact it, if that makes sense.
Okay.
Cool. Then I briefly stop sharing my screen. And then we have a few minutes now to kind of discuss what we've learned so far, what you guys think. Is there anything that's where you're like, that doesn't make any sense to me, or you're all happy with what we've done so far? So if you just want to kind of, I think for now, if I stop sharing, we can turn on the videos for like 7 minutes and discuss what we kind of think of everything so far. Right, anyone having opinions on what the data look like in 3D? What do you think about looking at the different of the voxel in like its X, Y, and Z? Do you think that was helpful? Do you think, no, actually, I knew that, no one cares? Do you think you have learned something so far?
Yeah, definitely. I think I got the point about ROIs, you know, how we analyze the background vessel and non-vascular for different fish samples. So I got it, yeah.
Great, thanks. Anyone else? Any thoughts?
I think I'm just waiting for the ROIs to make sense after we process it, right?
Probably, yeah.
I assume.
Do you think the kind of looking, for example, at the data with like the different voxel properties, was that important for your work, or do you think, no, actually, who cares if my voxels are X, Y, and Z, different or indifferent?
I mean, for my purposes, I'm always taking like my Z, like it should be similar Z stack style, so they should not be changing according. But I mean, I could see that being an issue, and I know I've seen that in imaging analysis pipelines where the voxel stuff definitely switches back and forth, so it does become an issue.
Cool. Anyone else? Any thoughts so far?
Well, in terms of sort of applying this formula to, isn't there user bias because we are selecting the vascular signal, we are selecting the non-vascular signal?
There's always some form of bias, yes. If you do it like that, you could do it on a slice by slice basis, etc. But I found that, especially when you choose the same vessel, it makes a lot of sense. And I'm going to show a few images in a second. If you guys just want to talk to each other and kind of just have a quick chat, and I'm just looking for 2 images which I can show you to why it makes sense, then it probably makes more sense to look at them.
I guess it would make sense to always save your original image as one file, and then as soon as you start processing, right, you want to make sure that you start adding stuff, because then your voxel, I didn't realize that like if you change that voxel setting that it would actually physically change it on your original file, like you couldn't go back. So that was something to keep in mind.
In a lot of places, an image, it has a link where you can click and say retain original, so it creates a copy as opposed to but I think you have to actively remember and click it, otherwise it'll do it on your original
having
copy.
that be the default.
Yeah.
Yes. Cool. So I'll just dig out these images for you guys to hopefully make more sense out of them. So this is one example where we look at different transgenics to measure the contrast to noise ratio. So for those of you who don't work with fish, transgenic is basically where we insert a certain fluorophore behind a certain promoter. So for example, in this case, it's different vascular genes. So one of them is a receptor, another one is like expressed in the vessels. And so we can use these different promoters, and they have obviously different fluorophores. And so using this, we can then assess, for example, contrast to noise ratio in different transgenics. And with this, we can then see this one, this first one, that's the one that we're looking at today, is actually the one with the best image quality in comparison to the other ones. We can also look at contrast to noise ratio over time. So in development, this is very important to see how these things change, especially because, you know, fish grow. So you want to look at that. And also you can look at how this is influenced by different diameters. So this is different diameters, but in this case we see that contrast to noise ratio is the same for these vessels, independent of the diameter. So this is one thing why we use measurements like this. And the other one is actually to look how processing impacts our data. And the next step that we're going to do is actually processing. But because Natasha was so keen on knowing why we do it, I just thought I'd quickly show this as well. So this is one measure. Can you see that? No, you can't. I copy that quickly into the chat because I think you can't see that right now. Right, so I've just put a PNG into the chat because I think it doesn't show on the screen itself for I don't know which reason. But if you click on that, what you see is actually the CNR in the original and the CNR in an enhanced image. And this is the next step that we're going to do. And what we do is we save these ROIs. And so we have first measured the CNR in the original, and then we measure the CNR in the same ROIs after enhancement. And what you see is that the contrast to noise ratio definitely got increased by this processing step. So this is our justification that we say this processing is working because it improves our quality of images significantly. Does that make Yeah.
Oh, I just have a question. Have you ever encountered a data set, or like where would you put the line at a data set that's not worth proceeding with because of the CNR? Like the CNR is too bad.
So like I said, fluorescence microscopy, we're really, really blessed. Like most of the signal is incredibly good. However, so one example that we actually see here, this one. So this is a construct which is incredibly great to image in the trunk, but in the brain we have a lot of signal from, for example, the skin and autofluorescence because we have to have a very, very high laser power. And so in this case, it's a fly-1 light-effect and clover line, which visualizes actin, so very low levels of a cellular component, so to speak. And so this one, it still has a fair enough contrast to noise ratio, but we see in the processing that it actually gets quite challenging. So already if you look visually, you see that here the skin has a very high signal. And so things like this, you need to be very, very careful, and you can obviously not apply the same approaches to this transgenic as you can to the third one, if that makes sense. So I think whatever you do, you kind of need to first understand if your processing is good enough for the specific experimental setup, if that makes sense.
For this kind of example where the skin is, I mean, this is something that I deal with all the time, is having issues with like the skin showing up lots of signal and then the brain not so much. So is this something that it's easy to segment out, or?
It's actually super challenging. So what we do at the moment is actually apply this workflow that we're going to talk through after this now.
Okay.
But then in the post-processing, we actually remove the skin afterwards.
Okay.
Because most of the time what we see is like that these signals here are normally not connected to the vasculature, and they're normally like small speckles, or they're like sheets, so they're not tubes like the vessels are. So we can use assumptions like this to actually get rid of it. But obviously something that is like this, very, very clean and clear, is way easier to process than, like you say, when you have issues with this, with the skin and autofluorescence, that's always very challenging.
Okay.
All right. Is everyone still all right? We're all on the same page. All right. I just stopped that sharing again. And then the next step that we do is now actually enhancement. And while this is kind of running, we can have a quick break. Again, it depends on your computer. If your computer is not the strongest, you can just skip the step, and I'm going to talk you through how, why, etc. So I'm going to share this. Okay. So if you go again to your data file, what we have done now is to look at these original data. This is the unprocessed data, which are just downsampled, but they had nothing done to them. And what we want to do now is actually to go into the processing, so this enhancement and improvement of quality. And what I said before, what we do here is actually use this filtering approach, which uses the assumption that your vessel is locally tubed. So these filters, there are multiple very famous ones. One of them is Frangi filter, and the other one is Sato enhancement. Both of them are implemented into Fiji, and there are actually two versions of Frangi and one of Sato. Whereas the Sato one is called tubeness filter in Fiji. I personally find this Sato enhancement to perform really good on our data, whereas the Frangi ones are not working that well on our data. But again, it really depends on your data and the properties, etc., that you work with. But these are, if you work with vessels, these are really nice tools to work with because your vessels are tubular. Same for lymphatics, they are normally tubes or nerves. They're like more tubular than anything else. And so in this step of pre-processing, what we assume, like I said, is that we have a tube. And to do this now, what we'll do is actually we open up the, sorry, I've got so many windows open. We're going to open up the macros. So if you have downloaded your folder for the individual macros on the GitHub page, and then have extracted your macros, this is what it should look like. And there are loads of them, but this is because most of the steps were, or I tried to supply individually. But in this case, what we'll do is just use this zebrafish vascular quantification graphical user interface one. So this opens up the graphical user interface that I've shown you guys before. And if you just want to drag and drop that, it should open up automatically. If it doesn't, it's probably because you have a Mac. No, let me know if there's an issue with opening it up, because you could also open it with plugins, macros, and edit, for example, and then open it up. So if there are any issues, just let me know. Otherwise, if you do get to the step, again, just give me a yes, or if you don't get to it, just give me a no help or anything. Great, thank you. I just start talking through, but don't worry, we'll all kind of catch up. So just so that everyone kind of is on the same page. Right, so once you have unzipped this, like I said, there are multiple different steps, and you can just load in this graphical user interface. And if you actually click on this run, it will open up this user interface, and all of the individual steps are implemented here. But like I said, today we just kind of mainly focus on certain steps because of time. But if any one of you has kind of questions on the other steps or really wants to use it for other things, please feel free to get in touch, because I'm more than happy to kind of help each one of you, because I think that's what we should do. And so what we're going to do is actually use this enhancement step. And so if we go to the practical sheet PDF thing on page 4, again, this is where we are now. So we can work through this step. Sorry. And so basically what we want to do is run this on the original data. However, if your computer is slow, or if you have any issues, like I said, I've run everything already. And so what you would see is what you will see is in your I2K data 512 folder is actually a folder which is called TF. You can, if you want to run it to see if it actually does run on your computer, you can first rename this folder to TF2 or test or whatever. So it's already in there. And so if you know your computer is really at capacity already, then maybe just work from this one here. But for those of you who still have capacity and the computer is working fine, you can go to your graphical user interface and make just sure that everything else is closed, like I just closed everything else, because we don't want to confuse Fiji, so and also don't want to use a RAM that we don't need. So always kind of have a fresh start, clean start. And then I'm just checking. Andre, if you're with, like, if you are at this step then as well, just let us know so we kind of know we're all at the same step.
Oh, weird. So I started running it and it's giving me an error, incorrect path file. So now it's saying, I guess that I don't have it. It didn't make a folder for the TF.
So it should automatically make it. If you go on, I just quickly rename my folder. So just to have it, because that's the original data, which you should never do, but I will do right now.
Is there a good way to like quit it, to pause it, and then?
You should always be able to just kill. So if you go on the kill switch.
Okay.
So if you now click run, and then what you want to do is So if you're very confident, you can click all of them. But I normally run each one of these steps, just step by step, just to make sure everything works and nothing is overwritten, and just take my time, especially because this tubular filtering takes as well. And so in this case, we want to select yes for the tubular filter, and we leave this so-called sigma size. Sigma size here refers to the size of the filter. And as I said, it thinks your vessel is a tube. And so in this case, it says, or what we said is like that the average of our vessels is about 10 micrometers in size. If you work with vessels which are 20, then you increase it. If your vessels are smaller, then you decrease it. But in our case, we set it to this 10 point whatever, because we found this to be the average size. And if you were to click OK, it should ask you for your input files. And in our case, this is the original data. And what you see, one thing that often happens is that you think you are in this main folder, but actually you are in one of these, which might have happened to you, Natasha.
No, I think I actually, because in the middle, I had already clicked run, but I hadn't renamed it. So then I think it got confused. There wasn't a TF file for it to go into after it had been started.
It should automatically write them.
No, I think, but I had switched the name in the middle of my run, right? Because you had already.
Okay, yeah. Okay, got it. Okay. Cool. And then, so yeah, if you click your data as an input, so these are the raw data, then it would normally just start to run. So this is one of the processes which takes a very, very long time, unfortunately, but this is because it's computationally very complex to run. You can let it run, and if you have light sheet data, for our data, it takes about 40 minutes to run on a 64-bit computer with about 128 gigabyte of RAM. So it takes quite a while, and it's quite computationally intense. And so you would see that in here to actually run quite a while. And how is everyone kind of doing? Andre, are we at the same point? Everyone still here?
Yes.
We can't hear you, Andre. You're muted.
So we don't need to specify a specific data file. It runs this tube filter for all the files in the directory.
So it will run it for the original data in the folder. So for all of these 6 fish that we looked at, and that the specification that we have coded here is actually for TIFF files, but obviously you could change it to whatever your data look like. So this is one of the things that we kind of customized to our data. And so then it would run it on your 6 data, or your 6 images in this case, which are in there.
So you run it on the TF folder or just the I2K data folder?
The I2K 512 by 512 data folder. So because the TF would be the output. So this is tubular filtering, TF, that's what TF stands for.
And it automatically segmented out the TIFF files, right?
Yeah.
Okay. How have you done this sort of I assume you have, for like time-lapse data?
I haven't run this on time-lapse data, to be honest, because I'm working more in like chemicals and completely different time points. But you could do it on individual time points by just specifying the input, basically. So if you have your time lapse, you basically just split it by times, or you could say you load the complete image, but then you just read every, let's say every 5 time points or whatever it is you're interested in.
Okay.
So yeah, we stuff like that is normally quite easy. The main issues with time lapse is that they're quite large, and so to hold them
and
on your computer,
it's gonna get
yeah.
Your analysis is gonna get compounded, right? So
yes. Okay, so most of us having or running it. Are there any issues for anyone?
It takes time.
Yes, it will take its sweet time. Have you all renamed the input folder, or have you left it? Are you just overwriting it?
Renamed.
Renamed.
Thanks.
I got my first one back. My first maximum intensity projection, so one's done.
Cool. How does it look? Good, bad?
It's the drug one, so they look like crap.
Yeah, I think what I've just seen is actually that I made a mistake in all of this, which is if I look at the maximum intensity projections, yeah, is that what you see? So this is one of the drug ones, that they actually have thicker vessels now. So what I should have done, because we have downsample data, is actually change the scale. That doesn't matter. Fine. So this is one of the things that I make obviously often as a mistake. Downsampling is not my friend. No worries, Andre. If you just want to, you can hear us, right? Okay. If you just, you know, kind of keep, or like, I don't know, just in the chat, then it's fine, so we kind of know where you are. If you are stuck at any point, just, you know, just type in the chat and we're kind of catching up with each other. If everyone else is happy, what we can do is take like a 5-minute break and get a cup of tea, comfort break, stretch our legs. Just don't listen to me for 5 minutes, if that's okay. And we'll just come back at like 19, it's my time, 19:53, so 5 minutes literally. I don't know your time zone, sorry. So basically 5 minutes, and we come back, and if anyone has issues, then we can sort them out. But just now have a quick break and just have a breather of all of this. Okay.
Before you go, one quick question. So we don't actually, the TF is the output folder that you already made for us. So we can move that folder out, right? We don't need that folder. Okay, got it. Thanks.
Just don't delete it, because we might, you know, if the other stuff takes too long, we'll just take on with that. But
sounds good. Yeah.
Yeah. Okay. Natasha, you're doing the right thing with the cat. Good idea. Cool. All right. So I'll see you in 5 minutes. All right. Okay. Hi, most of you, not all of you yet.
Hi.
Hi. It's nice to actually see people.
Yeah, I actually got a macro error. I can send the image to the chat.
That would be great.
It says Hessian AGN values only for 2D data in line 282. I'm going to send the image.
That sounds like it's run on the wrong folder.
I selected the general folder, the I2K data 512 by 512.
Okay, you can try and just start rerunning it just to see if it works. However, so this is why we just load in 3D TIFFs, because if it just runs on 2D data, it won't work, because we have this assumption that it is a 3-dimensional tube. And so basically the error that you've got is that it isn't able to extract these eigenvalues or Hessian matrix. And basically these are the ones, just share my screen again.
I'm trying to save the image.
That's right. So these are the ones where we take here. So the Hessian or the eigenvalues are basically describing your data in 3 dimensions. And what it tries to do is basically to say everything along your tube has the same intensity and therefore the same eigenvalue, but everything that is outside your tube will be having a different kind of data structure. So it needs the 3-dimensional information. But you have renamed your folder as well, right, to TF02 or something. So you didn't overwrite the other one.
No, I changed it.
Brilliant.
I'm going to kill and try to do it again, I guess.
If you want, yes, but it's going to take some time. So I'd say we'll just wait until everyone is back, and then we can discuss kind of the next steps. And if you haven't run it, or like if you haven't overwritten your TF folder from before.
No, I still have it.
Yeah, then we can just run it on that.
Okay. Thank you.
Welcome. Natasha, your cat makes me incredibly happy because it's really nice to, you know, see some
He's a
private life.
relatively friendly cat when he decides to be. I don't know if he's happy about work from home or not, but
Also have a visitor here.
there you go.
Oh, bless, see, that's great.
You can visit me now. Take your butt out of the camera. Come on.
That's what we need nowadays, kind of sharing the good things. I'm just looking at the macro error what it says. Did you try and rerun it if it works?
I got distracted. I'm going to do it
No
now.
worries. Oh good, oh good, oh good. I'm just interested to see what the error was, but if it works for everyone, fine. And I hope that it's not the code. Hopefully, we'll see.
So far, mine's been working. I've gotten through 4 images so far.
Right, same from yeah, I'm on the 5th now, so good. Right, I think most of us are back from the break. Brilliant, let's see as well. Jin Yun, are you back as well? And perfect. So I think it's almost all of us back here. I hope you had a nice break. All right, sorry. Cool, so this is still running, but we can talk about the next step, which is the segmentation. And in this case, like I said, we use a very simple thresholding method. So this is now in the PDF, it's basically done on the page 5. But before we do this, we actually kind of want to look at how the data look like. And this is where we would normally, on page 4, at the bottom of the PDF, look at the contrast to noise ratio. And this is what I've shown you before, which is basically this quantification, where we look at the original data and the enhanced data, and this is where you then can quantitatively assess that this enhancement should have hopefully improved your signal in the vessels and decreased any non-specific signal, especially in the brain of zebrafish. There will be some autofluorescence, especially some transgenics are very, very prone to having a high autofluorescence background, depending on their wild type background.
Have you found that that's more true for like GFP than for like a red fluorophore?
I think it's less the fluorophore than the actual construct. So, for example, with this M clover construct that I've mentioned before, we have this extreme, so we have to have a laser power of like 25, which is just ridiculously high, and so you get a lot of this background which is kind of unnecessary, or like it's just there, well, but not really informative. I think it also has a lot to do with the actual background of fish. So if they're in fish, we have different kind of wild type strains, so A, B, WTs, they're all like different breeding backgrounds. And some of them, like LWTs, they have a very high autofluorescence. So I find that sometimes a bit of an issue.
Can I ask an obvious question?
There's no obvious
question. I mean, CNR calculation, like when you calculate the mean, for example, for vascular signal, do you take the mean of means, like of all the layers, or you find the mean of an appropriate layer? Like,
so in this case, I just take a single slice.
Single slice, I see.
Yeah.
So which is most intense, I guess, right? The most intense slice, you take the mean from that. I see.
So I take basically the slice which is in the center of the vessel, because the vessel is like this circular thing, and where I see it's the highest intensity, I basically take this. But obviously in fish we have like, so this is now just an N of 12, but normally you'd have like N of 20 or something. Go ahead.
Okay, and how do you select the slices for the non-vascular signal and for the background?
So this,
are they the same slice, the same exact slice?
So I just open a second instance, but yes, I normally tend.
Sorry for backtracking.
No, but that's exactly what we're here for. I really want everyone to kind of learn something from today, and no matter how small, large, or whatever it is, I think it's great that we have this time to discuss all of this today. So if you'd choose your image and then go through it, and then I should have this somewhere.
Mine just finished. Everything ran smoothly.
Great, that's good. That's what we want to hear. So if you go to your vessels, like I said, we normally use this standard vessel as kind of the reference point. But again, you could do it at other ones, whichever you find are most reproducible. And I appreciate that there will be data where it's not possible to use always the same one. But basically, if you go through it like this, you will see that about at this point, it's probably halfway through this three-dimensional region, and this is where I pose my region of interest. And it would actually take then the non-vascular region, for example, here, because there is just no vessel, so it's the same size, and then same for the background, and go into it, just to kind of avoid having to scroll through everything, especially if you have to have, you know, hundreds of images. It takes just a really long time, so I tend to just work on the same slice. There are some steps how you could do it in 3D and make really a nice sphere, etc. But like I've shown you before, that the main thing is, if you want these comparative kind of examinations like here, we found that it's enough to have one region of interest per vessel, basically, or and then compare it. And when we applied it to several regions, etc., we didn't find a difference. But that again might be different on your data type.
Okay, thank you very much.
More than welcome. Any other questions at this point?
So it ran for the TNN2 NNT, the treatment, but the controls, it showed an error and then stopped. So I had the max projections and the enhanced files for the treatment, but I don't know why it stopped for the uninjected controls. I think it may have a similar error to what Mariana was saying. But it's okay. I have the enhanced.
Can you just share your error code as well, because it shouldn't throw up these errors. So, but it's good for me, because you know that the more feedback we get as developers, the better it is, really. They sound annoying, but they're great for future users. So yes. Yeah,
I closed it, and it won't show it to me again. Even when I say show errors, it's not showing it. So if I run it again, I can send it to you.
Great, thank you. That would be really appreciated. Right. So for most of you, we still have the original TEF folder. If you haven't got it, let me know in the chat. Okay, Naka finished, that is great, finished too. So most of us are finished now. And so what you can do is then actually also overlap your data. And I find that very, very helpful, because I'm a very visual person. And so for me, kind of overlapping images, etc., is really something very useful. So I opened the original in this case, and it's like an uninjected control. I'm just kind of giving you tools. You don't have to do everything, obviously, but I personally find this something very helpful. So if I've done a processing step, I can just basically drag and drop this into here. And so in this case, what I see is that this is a 32-bit. So it's a tubular filter, this enhanced one, but it's now a 32-bit one, whereas the original is 16-bit. So to actually compare them, I just make them both now 8-bit. You could do them all to 33 or whatnot, but we just need basically to have them more or less the same. And then you could go to color merge channels and then overlap them in different colors. What I realized is actually that I think I've run all of these with a different scale size, because obviously I did this this downscaling and I'm really not used to it. So I'm very sorry for this. But this is life, right? So what we see if you do this image color and merge channels, sorry, I went through it quickly, you can select these different colors for the different channels and then create an overlap. And what you should get is something like this, so an image with all the different slices, but with two different channels. So one of them would be the enhanced and the other one would be the original. And you can actually assign them different colors with the lookup table. So if you go to image and lookup table, you can go to, for example, green, and the other one you can set to, let's say, magenta. And then you can actually see how they overlap. And I find that something really, really helpful. And you can kind of, if you zoom in by going onto this magnification glass and then just zoom in, you can see where it performed well. Like here, you see that they really overlap very well. And you see regions like here where it doesn't overlap too well. And again, this is probably because I've set the wrong scale, but yeah, it was very late when I did that. So apologies. But I find this something very, very helpful, and do this literally after most processing steps to see what makes sense and what doesn't make sense and what kind of needs improvement, especially because enhancement or pre-processing can really be the make or break of any segmentation that follows. So spending time on understanding this is really a crucial thing. Okay, now I'm just closing this and just checking my other, sorry, my other instance. Where are we here? Okay, this one, so mine is now finished as well. So what I get is this macro finish, which is always a nice thing if there's not an issue. Are there questions at the moment?
So you keep saying that it's because the scale wasn't the same. Where was the scale not set?
I have, so because when we go to the, if we run this here, you hear this, you can change the scale. And I optimized this 10 for the original data, which is this 2000 by 2000, but we actually do run it right now on 512 by 512. So they're slightly over enhanced, if that makes sense.
Okay, so the enhanced ones they do look.
Yeah, like blodgier than they should do
okay.
in reality.
Okay.
If you wanted to run this similar kind of pipeline on your own data set, so for a folder, what kind of material does that folder need to create this kind of TF images for your own data set?
It basically just needs your original data. This is what we've done. So we run it on these original TIFF files. So like I said, in our case, I've implemented it to read in TIFF files. But because we do use a light sheet microscope size 1, the first step that we do is actually CZI to TIFF conversion. And so if you'd have like a LSFM or CZI or whatnot, you could change that. But I'm also happy if any one of you have like issues like that, I'm more than happy to just change that code for you guys. Same with like single and dual color. At the moment, it works on 2 color images. So it will split off the red and green.
Mm-hmm.
Yes, thank you. It's now gone 8 o'clock, and now I'm like, okay, I'm realizing. So you could change all this, but again, if one of you says, oh, actually, I have a 10 color image or whatnot, I'm more than happy to help you guys to just set up the thing. The actual thing that you need is a TIFF file to start with this tubular filtering. And then one thing is obviously kind of optimizing the sigma size. So we found, for example, in mouse data, that this definitely needs optimization, and also potentially being run on multiple different scales. So this one should consider different scales, so small vessels and large vessels. But in mouse data, we have such a huge range of vessel scales that I find it good to run it on like 2-3 scales and then really combine them and look at them and spend time on finding out what works on this kind of stuff.
So you don't. So I guess I got a little confused. I thought that the CNR that we calculated, that it needed some kind of, to normalize, it needed some kind of background information. So all it needs is the original TIFF file, and in this case, we are specifying the sigma size.
So this is for the tubular filtering, yes. Where we need the sigma. For the CNR, we basically normalize it using this non-vascular region.
I see.
Does that make sense?
It does, but is that being applied in the tubular filtering or no?
So when I showed you the image, this one, yes. So I ran it first on the original. So that's what we've done before. But because of time, I'm not letting you run now on the enhanced ones. Normally we would do this, but I'm aware that we're kind of falling slightly back on time. That's why I've just showed you. So normally this would be where we now do the same CNR measurements in the same regions of interest. This is why we've saved them on these enhanced images. So you can compare them to each other.
True, but the CNR is just a metric to see how good your signal is to your noise. But when you're doing the enhancement, you're not using the CNR values. That's just a test of did you actually enhance your image or not. Okay,
yes, sorry, yes. Right, I think most of us are finished with the macros. Right, let me just check where we are. So yeah, so normally we would now quantify the CNRs, but I think we're slightly falling, we're slightly falling behind at the time, so kind of skip that now. We kind of go now to the segmentation, which is this binarization process. And like I said, here's also where we extract the region of interest. You can ignore that if you're really confident in your data, but I normally really exclude, for example, the eyes, or if you have whatever your data are, you kind of really want to make them comparable to each other. And so we're excluding certain regions. So for example, in our case, we know the eyes are not of interest, and because everything is basically off that's beyond this certain point. Again, there are loads of different methods to actually do a segmentation. And this is something where if you do data analysis and want to analyze your images, you should spend the most time on this step, because it can be really, every data type is different. It can be different if it's confocal light sheet, or even between light sheets if it's a lattice vessel beam or a standard dual site illumination. It can be really, really very different depending on your data types. But in our case, we use a very simple method, like I said, which is based on just your image histogram to make these two clusters, namely vascular and non-vascular, to binarize your images. And again, now we would manually draw a region of interest, which I just quickly show you. So what I normally would do is basically open my TIFF file and then just literally draw around my fish. So sorry, just color lens. So I would, for example, say, I know this is my region of interest, or you can also make it as a stack or like as a maximum intensity projection and draw it around. And similar to what we've done before, I just draw something like this now. So this is my eye or my head. You could edit to your region of interest manager. Here, just trying to move this. Your region of interest manager, and then you can save it again as embryo 1, embryo 2, again, to save time, I have actually made this folder again for the ROI sets. But if you have time, you can always kind of play around with that. And like I've said, I will be there on the Slack channels. I'm there by email if you have any questions whatsoever. So I just delete that for the sake of it. But again, you can just drag and drop, or if you're a Mac user, go to your region of interest manager, more, and then open. And what it should do is open basically these 2D region of interest. Again, if you have the time, you can draw 3D regions of interest. But I personally
folder. What? Because I have the original CNR ROIs, but where's, I'm not seeing the ROI.
It should be in your I2K data 512 by 512, and it should be after the README ROI set. If not, I can just copy it in the chat.
Yeah, no, sorry, I was missing it. It was zipped. It's my fault.
No worries. Okay. I'm just checking here as well. Okay. Right. And so again, I've drawn it for you, but you can obviously, as you see, they're really just crude for the tutorial, but obviously you can draw them, you can change them, etc. And what we'll do is we'll use these regions of interest actually now for the segmentation to extract these eyes and regions that we're not interested in, basically make the fish more comparable.
Does it matter what you name them?
The only important thing is that they're basically in the order of what your images are in the folder. Because in Fiji, basically, it opens with the first instance of your image in the folder. And I've written it so it runs on the whole folder just to make it quicker. But you could literally, you could call your fish, I don't know, Henry, John, Peter, you know, like that wouldn't matter. It's more like how are they ordered in the folder itself.
And does it matter that we have all those original ROIs like still in our ROI manager?
So yeah, for the next step, what we'll do is we close everything again, because so again, Fiji starts always at the top, which is good, but it can be annoying. So with stuff like that, always kind of start afresh. And so basically just close everything that you don't need. I, for example, have 2 instances of Fiji open, which I really don't need. So when we have that, the next step is now going to work on the actual enhanced data. So basically we had the original, then we had the enhanced ones, and now we segment them. And so again, if you would go to your actual data in the TF folder, what it should already have is actually fresh order data, which I actually want that. Well, okay, I probably forgot that somewhere. Oh no, wait, sorry, I renamed it. There we go. So if you have renamed your folder and look at the TF02 or whatever you called it, you should have a folder which is TH, which is thresholded or segmented. This is what we get now in the next step. So if you want to run it on the ones that you've just done, you would take your TF folder as an input. So basically what we do is we choose segmentation and say yes, and we say a ROI set exists. So this is the one that I've shown you to draw around the brain. It would give you different options to whatever you want to do. Sorry, I'll just get the Slack stuff as well. So if you say no, it would give you different options, but because of time, we don't run through everything just now. And same with like, do you want to perform downsampling? No, because our data are already 512 by 512. But at this step, I would normally just downsample them, because the later steps, such as the skeletonization, just take a very long time, and it doesn't really make a difference if you run this on the whole data or the small data. So this is where I normally downsample it. And so I would say yes, and then if it asks you again for your input folder, and like I said, in this case, you would choose your TF folder. And if you said yes, then oh no, tell me where the ROI set is. Sorry, that as well. And in this case, it's in the I2K data. Everyone still following? And then say select, and then it opens these ROIs automatically. And then it should just run this segmentation. So in this step, like I said, it's going to do this auto thresholding. It's going to select the edges. So what you see here is already the edges of your image. So basically this is... Mine's
having issues trying to run, like it keeps giving me the ROI issue that it doesn't have it set.
Do you want to share screen so we can have a look at it, or I don't know?
Sure.
I can stop my screen share.
See how messy everything is. Okay.
You guys have seen my screen, so we have nothing to hide from each other.
Yes, segmentation. Yes, ROI set exists. Okay. And then I'm asked here, and there's where it is, right? So I was just going to say, okay, open.
But that's in the max folder, sorry. If you can just kill it.
Yeah. What was the issue?
Just say yes and yes, and then just look at the folder again, because I think you've run it on the maximum intensity projection folder where it says max. So yeah, I can point. Wait a second.
Because I'm just in the I2K data folder right now.
Yes, but this one here is maximum. Have you chosen this one, or have you chosen this one actually? What sometimes it happens with Fiji is actually that it tells you that it chooses this one, but it actually attaches something on the back. So what you can do is just go one folder back.
Okay.
And then.
Yeah, but see, I still get this error.
Oh, because you selected now for the ROI set. Can we just do it slow? Sorry. Just one step at a time.
It's not giving me anything telling me what I need to be selecting. So it's not prompting me for what I'm selecting?
Oh, it should do. Yeah, it should say choose your input folder, which is in this case the TF folder.
Okay. Okay, so now I say open that, and then now I'm also still not getting anything.
And now it should tell you that it is selecting the region of interest. And I will actually change that because I never knew that it doesn't show you on Mac, which it should do. Yeah. And then don't click without, and just in this folder, in this I2K data folder, it should be in there. If you can just try and run it.
Okay, just hit open. Okay, now, okay.
Cool. So it was just an issue with the input folder, but it's really useful to see that on Mac we've got these issues. So I'll make a note of that as well. So this is
yeah, because it's not telling me which ones are which ones need to be input when I'm selecting my folder.
Does it automatically save to a TH folder? So will it overwrite it?
Yes, it should do. But again, you can change that, and you can have a play with all of that. If you guys are actually more interested in all these steps of the GitHub that I've shown you, where we downloaded the individual macros, it actually has like an idiot's guide. So it's more written for myself than anyone else, but it's literally like a choose this folder, choose that folder, this is what you'll get kind of thing. Because I'm very, I'm working quite a lot of projects, and so I always tend to forget very easily. And so for tutorials like this one, I go back to these workflow documents, and then I'm like, oh, this is what I choose, this is what I do. So if you guys want to use this user interface, there is more documentation on the GitHub for all of you if you want to.
And if there wasn't an ROI selected, like if we didn't have those ROIs selected, will it prompt you manually to do it for each image, or?
It will give you different options, and now I don't want to lie and say anything. So I think if you can say you can draw them before manually, so you can also draw them, or you can also stop after the segmentation in case you want to do it. Okay. When I had like an earlier version, I think what I did, I was just drawing a massive like a rectangle around everything and just running it on that as well, and just being like, let's see what happens. So that stuff works as well as kind of workaround, but you can run it just to do the segmentation as well.
Okay.
Okay. Right. So have the segmentation, has it stopped for all of us? Haven't heard anything from Octavio, Alpha, Chiara, Tim, Yun in a while. Liz, are you all still there with us?
Yeah, I am trying for the second time. It was working. There was a point it stopped, I think. So I am trying for the second time.
Did it show macro finish, or did it just stop or like break or anything?
No, it stopped. It says index 6 is outside of the 0.5 range in line 402.
Ah, okay. Have you closed your region of interest manager before?
Maybe. Yeah. So it's open now.
Okay, you should close. So that's kind of the issues that come up with the region of interest manager, because unfortunately not very straightforward to code it into anything. Mm-hmm. So this could be if you have too many regions of interest open or too few regions of interest open, because it just takes literally the ones that are in there.
So I will restart Fiji then. Right.
You can just close your all the images and your manager, and then you should be able to run it again.
Okay. We don't select downsampling, right?
Yeah, because that's like I said, like we've done before. So I've provided you with downsampled data, so they're already smaller.
Okay.
But this is something, especially if we work with light sheet data, at some point you kind of just want to downsample them just to make them smaller. So at this step, I would also say, for example, you can run your segmentation on your unprocessed data, but because of the sake of time, I actually just show you the results basically. So I just close everything that I don't need again. And so if I've basically run it on the enhanced images, and so what you get is this threshold data. And then again, they shouldn't be looking like that blown up. Again, my apologies, but that's all we have today. And so this is what your maximum intensity projection of your threshold of data look like. If you would drag and drop it into your Fiji, what you see is that this is now your binarized image. So you only have 0 and 1 or 0 to 255, nothing in between. And this is what we can use to quantify the volume and the surface, etc. If you would run it without the enhancement, you would actually get something that looks more like this. So it can't cope with it. For example, this one is a nice example because you just see it's really not able to distinguish background from the vessels very well, especially in the fish which have been treated because they have a lower image quality themselves anyway. So you kind of get really bad data. And this is really why we use an enhancement to improve our image quality. And so in your tubular filtering folder that we've done now, you will also have a folder which is called edges, which is basically the surface of your data. And in this case, this is basically the outline of your object or your vasculature, so you can quantify the surface of how much surface you have. And it will also give you this text file where it actually quantified the vascular voxels and the vascular volume, as well as the edge voxels and the edge volume. There are two measurements because, like we've learned, voxels have also volume. They have an X, Y, and Z direction, so they have an intrinsic volume. And so if you image one data set with a different size of voxels, you want to consider that, and that's why we always kind of have two measurements just to look at them. So the volume should be comparable between your samples because we normalize for this difference in voxel size, but kind of just to always have a reference point, we also look at the voxels. But to quantify things, we normally just use a volume because it's more intuitive for a human to look at volume measurements. Okay. Okay, right. Then, does anyone have questions on like the image segmentation, or does the segmentation make sense to you, or all happy? Okay. Moving on from this, the next step would be to again run it, but now actually look at the quantification. And this is where I said, or where we now quantify the length using the skeletonization as well as branching points and diameter, just to kind of give you a reference to what I'm talking about again. And the skeleton is where you extract your center line of your vessel, and in this case we use the skeletonization or the 3D iterative thinning in Fiji. Following this, we then look at the vessel diameter using the so-called Euclidean distance map, which looks at the thickness, and then we get this one voxelized representation of the data. And all this is being done inside the software. But because it's a macro, all of you can look at the code, how it's being coded. So if you want to kind of use sections of the code, feel free to do so. And as long as you kind of just reference that the code came from us. Right. So the next step, like I said, would be this number 7, which is the quantification. And if we here go to select this yes, and then say, are the data downsampled? So yes. So at this point, if your data wouldn't be downsampled, it would actually ask you to please downsample them because it otherwise takes long. Obviously, you can run it if you have time. Feel free to do so. But I personally I'm very impatient, so I like to just run it on downsample data. If you run in a new data set, which you have never run before, it's always worth to run it once on the data that are not downsampled, and then run it on the ones which are downsampled, just to compare and make sure that there's really no difference between them. We found that there is maybe like a 5% difference, or in worst case 5%. So it's normally kind of negligible, but just make sure that it is. And then in this case, it would also ask you about registration. This is something that we haven't talked about today because of the time, but this is where you would bring your embryos into one space according to the system to overlap them. It's especially helpful if you don't want to draw your regions of interest, or if you just want to compare your embryos to each other. You can do this registration and then run it on that. But for today, we haven't done it. So yeah, basically say just yes and yes, and then it should, so for you Mac users, I will try and get this where it says like input folder, because I've seen now on your screen that it actually doesn't tell you what we're looking for, so I'll try and code that into it. And so what we'll do now is first, again, choose the input folder, and from like the logic, what we've done is now we choose the thresholded or segmented images as the input folder for this segment, or for this quantification. So we can go into the I2K data, into the tubal filter ones and the thresholded ones, and select yes. And then it should just run automatically. And then again, it just shows you everything. And you can run it in a so-called headless mode where it doesn't open things, but I just left it because I like to see what it's actually doing. And so it should run like this. Just again, if there are any issues, just let me know in the chat, and we can solve it. And I share my screen again. So that should actually be quite straightforward. And if you look at, so if you go to your TF folder and inside your TH folder, it has made a new folder which is called analysis. And this is where we put basically all the output folders or output files in. It's still running here, so I'm trying to open a second instance as well. Right, so that's the second one. So if you drag and drop them now, for example, here, this is like a skeleton, and this is this one voxelized representation or center line that we talk about. If you look at one of these, then this is these thicknesses where high intensity is thick vessels and low intensity is thin vessels. And then these ones up here should actually be the 1D representation. And this is where we perform the diameter measurements and quantifications. And also these ones are saved in a way that if you actually hover over it, it should give you the diameter measurements. So you can, if you find a vessel that you're particularly interested in, you could go like, oh, like for us, this is one of the main vessels. This is the vessel which feeds into the brain. We can go over it and say, okay, this has such and such diameter, and etc. Or this is changed in a certain treatment in a certain way. And so this is basically for representation. But then also it gives you these files with the diameter measurements, etc. that you can then actually basically plot. And then it should be more or less okay. So yeah, that's done. And so this, what you actually see on the screen, is giving you the summary of the skeleton, which is why we downloaded the neuroanatomy one, because this would go into analyze skeleton. In the standard Fiji, you have the analyze skeleton, but what we did is summarize it as well. And so you've added that now to your Fiji with the neuroanatomy. If you have issues with this pruning of the skeleton, if you go to analyze skeleton, well, obviously something open. You can actually set your pruning here. So this is what we spoke about before with like how you prune it. I find like that the shortest branch pruning works pretty well on the data that we work with. But again, all of these kind of steps, have a play around with them and see what it does. Some of them work really well, others will just crash your computer, etc. So you will see it doesn't work. So that's kind of a normal thing. Right. again, I just close everything that we don't need for now. And if you can just put in the chat if you're running, if you're having issues, that would be great, because yeah, a bit hard to gauge from here. Right, I think most of us are there. If not, again, don't worry, we'll just take it slow. Because at this step, most of the processing has run. And so here's where you'd actually go into this visualization. Like I said, this is kind of where you're now trying and see what works and what doesn't work. You have these different folders basically with your diameter measurements and your network length. So in this case, it would give you the TNS2, so the ones without blood flow, and the ones which are uninjected controls. And so it would give you the differences in diameter as well as, for example, the vascular length. And so this is also kind of the step where you try and see what makes sense. Again, you can plot all these data, and actually it left some time for all of us to kind of just plot it and have a look at it. But I'll just very briefly show you on whatever slide it was, this slide. So, because like I said, you sometimes really don't know where to start or what makes sense. What we found, like I said, is that measuring in volume is one of the kind of obvious things, and it's quite easy to do. So we do this on these thresholded data, so the segmented ones. And like I said, we use the microns, not the voxels, because microns have this 3D shape. After this, we have the surface, which is this edge detection, and we see that there is a slight difference between the fish. But it becomes actually quite interesting when we look at density. So how many vascular voxels are in the brain, because what you see here is actually that the density is not changed in the fish which are healthy against the fish which are controls. And this is because actually their brain is so much smaller. And this is something that you kind of need to consider as well. So how much normalization do you want? Because if we would, for example, do a normalization and normalize these brains to the size of these healthy fish, we would have a significant difference in density. And this is kind of where you need to understand, for example, does your fixation have an impact on tissue shrinkage? Does your antibody have an impact on the background, or how much is being wrongly visualized, etc.? And this is really kind of, once you get these data, as you can see here, you can make it really a judgment call on how good or bad your data, but also your quantification is. And so to actually start implementing a code like what we kind of run through today, I tend to use an example like what we ran through today. So these fish which have no blood flow against the ones which have blood flow, because it's so significant and we know that there's a difference. And so with this, we can then say what actually does make sense to quantify. So for example, branching points, we see that they have significantly less branching points. So we see that this quantification makes sense. And from what we see visually to quantitatively, we see that this is true, or as true as possible, basically. Same with the network length as well as the radius. So just by looking at these, we see that these fish have a collapsed vasculature because they've never had this blood flow. And so therefore, we assume and we can now quantify that these have this reduced radius. But again, I think especially the density just highlights how much you need to kind of consider your biology in terms of your data analysis, if that makes
Where
sense.
are you getting like stuff like the radius and the vessel diameter? Because
So this is in the last folder. So in
last one. Okay, it's not in this most recent analysis.
It is in this analysis. So where you go to, so it's thresholding analysis, and then it gives you diameter, network length, and this one here should be your diameter. And again, normally it would be significantly different, but it's just the wrong scale, and I really can only apologize for that.
Okay.
So this first column is diameter, the second one is network length. Okay, does that make sense so far? Yeah? Any questions? Okay. Right, so normally we kind of plot everything now and kind of look at it as such, but I think what we can actually do is start a discussion with what we've done, what makes sense, and what you guys are working with, and what you've learned today, and how you could apply to your data, and kind of maybe have a discussion of you know, kind of find solutions together, because I think that is also an important thing, because obviously all of you came today with an expectation to work with your own data. So I think that is important to do as well. So my question is now, do you want to have a quick 5-minute break and get a coffee or a tea, or are you good to go? Or what is your, so we've got until my time, 10 o'clock, so that's 1 hour, 11 minutes to now go through everything, really think about what we've learned, why is it important, etc., etc. Up to you, really. If you just say in the chat, like, break, coffee, help, or fine to go, just let me know, because it's very difficult to not be with you in a room, please. Right, okay. I think tea is a good idea. We should do that. Okay, should we say 5-minute break and then come back at like 5 to 9 my time, basically 5 minutes of your time, come back refreshed, and all of us then have a discussion together. Is that okay? Brilliant. Okay, see you in 5 minutes. Great, thanks. If you're all back and just want to write some notes of like what we've done, so these prompts we thought are basically really kind of to start off the discussion, because I think that's the most important point of today, to really kind of re-emphasize what we've done and why is it useful maybe to you guys, especially because all of us come from a different background. It would be good that all of us kind of learned something today. I've learned that there's a massive difference between PC and Mac, so I need to change that.
I mean, if people aren't going to come with, okay, yeah, macro stuff for my own analysis, yeah, I agree with that. I was just thinking I would, for, like, I know we use the, I can't remember what the smoothing was for the enhancement for the tubular structures, but if you were, would a cylinder still be counted as a tube? Is that enough?
Yes, so everything basically that's round and long is fine. The main distinction in all of this is really if it's a circle, a sphere, then it's called, in computation analysis, it's called a blob. And this will be different, and the sheet, because a sheet is flat and round. And so this is basically where we can extract, for example, the skin of the fish. All right. Any more comments or notes?
So with the way that we did it, though, the skin would automatically be segmented out because it's not tubular?
Yes. So no, actually we would segment it, but I can actually show you how to get rid of it as well, if you guys want to. If there are no more comments on this, I mean, I'm very glad if people say they want to use the macro, and if they say it's very helpful, that's good. Great, Euclidean distance map. Okay, so we can talk about this as well. And then taking individual steps of the macro. Sorry. So I just stop this here for now, and we can start kind of discussing things. So firstly, if it lets me stop. Oh, I need to get rid of the annotations first. Oh, there's another one.
Can you get rid of them, or do people who write them have to get rid of them?
You can get rid of them yourselves. So if you have this panel,
yeah.
See, I love this function. It's great, isn't it? So if you have this panel where you have mouse, at the end you have clear, and then you can say clear all drawings. You can say clear my drawings, or you can say clear viewers drawings. So you can have a play with that.
I guess if you're the host, then. Yeah, I can't clear anybody else's drawings, which makes sense.
Okay.
I think the host whose screen is being shared can decide who they want to clear.
Okay, that makes probably makes sense. Yeah. Brilliant. Okay, cool. So we have some things for discussion. So that's good. Let me clear this now. Clear all drawings. Right. So Okay, sorry. Right. The first thing we can discuss about is actually how to get rid of non-vascular signal. So we have at the moment some stuff on bioRxiv if you're interested in. And so this particular one looks at actually how to segment and how to validate your segmentation outcomes. And right, we ignore all of that because it's not very nice or not very interesting. So one thing was, for example, in this one, we look at the different scale sizes and the different filters, etc. And this is how you would kind of assess and prove that your sigma of this tubular enhancement would be appropriate for your data. All of this we can ignore. Sorry, just jumping. And then this one is probably the most interesting one. So this one now looks at really challenging data. Like I said, the magenta one is the one that we looked at. So this has these really nice contrast to noise ratio, very little background. Looks really nice. It's computationally not very challenging. But then we actually look at quite challenging different transgenics. And what you already see is that all of the green ones, we see actually the skin, for example, in the eyes, or here the skin of the body being already very visual in the different images. And you see that this really gets worse in the different lines, but also what you see if you look at these just here is that actually the signal that you see in the vasculature gets less and less and less because there's so much background. And so this obviously is more and more challenging for the data. And this is obviously something you don't always have perfect data. So we often work with data which look more like this rather than this. And so one thing that we start to look at is how does our segmentation kind of perform on this? And so what you see here is actually that most of the skin, for example, does get segmented. And so to get rid of this, what you can do in Fiji is use, for example, a connected component analysis. So if you go to your Fiji search function, you should already have connected components labeling, for example. And there you can examine if they're connected. So if you're for example, also in antibody staining, so you have a lot of times really these speckles, and to get rid of these, you can do this by connection or, for example, by size. And so you could do this in this step here using connected component analysis. And
if that's not popping up in my Fiji, is this just something that I need to update? Or
let me just see. Morpholib, oh, that's why probably. So that's actually a really handy tool to have. GitHub, you should have this workflow documentation, which is this 18-page monster piece where I said before, it gives you the exact input, output, and everything, the individual steps. But it should also give you this link to this Morpholib J plugin and the link to this one, and that is actually, sorry, which does the connected component analysis. So if it doesn't come up, you can download this one, and this is actually really handy because you can compare different images to each other, etc. Oh, thank you, Liz. She just said you can also run the connected component analysis on GPU if you have downloaded CLIG or CLIJT. If you have a good GPU, it may help speed up analysis. Really good point. Also, CLIG has a tutorial, which is great. But unfortunately for my Lightsheet data, I can't run it on my GPU at the moment because they're just too large. But if you have smaller data, yes, running it on GPU is definitely also an option to do. Okay, did that answer that question? Or do we want to talk more about unspecific signals or anything?
I mean, I can't use it right now, right? Because I have to still download the plugin and then reinstall stuff. But I think I'll have to go through and look through how to actually implement the connected segment.
And for example, size inclusion works or size exclusion works very well because the vasculature is a connected system. in humans, it's a tree, in mouse, it's a tree, in fish, it's an enclosed system. So it's very nice. It's the same for most neuronal systems, probably not for glia or astrocytes, but most systems are connected in some way. So for example, for glia or astrocytes, what you could do is also, again, a size inclusions, for example, to get rid of any speckles or background, and just say you want to look at in a certain size range, if that makes sense. Right. Then the other step, or the other question was like taking steps out of the macro that are useful. Again, you can refer to this workflow documentation, which is on GitHub, and again, it gives you each single step, how long it takes. So it has really way more information than what we kind of did today in 4 hours. It takes, so it gives you purpose, options, input, etc., for each individual step, and also what the output is. But also what we've tried to do, as you can probably see here, is that each step is very modular. So you can just run one single step at a time. So if you say for your case, you just want to run the segmentation, then you can just run the segmentation. If you say you just want to run the quantification because you have already segmented data, then you can just do this. If you have questions or if you want something specifically implemented, please really feel free to contact me. I'm more than happy to help if I can. If I can't, I will tell you, but if I can, I'm more than happy to spend time on really helping you getting things going. And so, yeah, with this modular implementation, if you just want to pick certain pieces, feel free to do so. Again, the paper for this is on bioRxiv at the moment, or the manuscript for this is on bioRxiv. Right, and then for the Euclidean distance map, so that I kind of have to be very, very transparent and say there's a huge debate in the field on how to measure your vessel diameters. There is a very kind of, what is the best way to do it kind of thing. Obviously, your vessels are a three-dimensional system, and so the perfect thing would be to measure, basically, if you have your center line, measure the diameter in like different positions and average it at this different position and along the vessel, etc., etc. But there is at the moment no option to do this in Fiji, and so this is why we went for this Euclidean distance map. If you, for example, say you want to test it against different methods, what I found, what you can do as well is actually measure thickness, local thickness. And so I don't know if that's actually a standard thing in BoneJ, which I don't have in here as well. Great, so if you go to your plugins and to update, there is one page which is called BoneJ, which is really useful. I can actually quickly show that. BoneJ Fiji. Because what it does is also it measures local thicknesses, and that's especially, for example, for Nagam, probably interesting, who works with skeletal data as well, to look at them in these terms. Obviously, skeleton or bone data are very different to vascular data, but it's a very useful tool. And what it does here, for example, it shows you the thickness. So in this case, what it uses to measure the diameter is an approximation by spheres. So basically, if we go to the slide somewhere here, you should all be able to see that. Sorry. So it basically uses something like this here. So it tries to use spheres and place them inside the vessel or the object or this bone. And based on how small or large the sphere can be, it basically gives you a color. So similar to the Euclidean distance map, the color gives you the thickness or thinness. But this is one other approximation that you can do to measure your thickness. But also, like we see on this figure here, so the red would be the center lines. So there are various different ways to do it. But in our case, we basically just use this thickness approximation. But again, there will be debate in the field of what works best.
But is like the thickness calculated on 3D images or 2D images?
So what I have done just for this graphic user interface now is actually to do it in 2D. So because it basically, so it is all produced in 3D, but the quantification itself is done in 2D just to speed it up, if that makes sense. However, you could also do it in 3D. It was just a question of time for me to say it's just quicker to do. But if you basically go to your Fiji and go to your data, and I just take some thresholded images now. So there's the 3D step. If you go here and then to process binary and then distance map, you do get this, or you should get this 3D distance map. Oh, I haven't inverted it, sorry. I need to invert it first. But if you invert it, you see then it works very quickly to do this distance map. Sorry, I haven't done this in a long time now. Which is the wrong way around as well. I'm sorry, I don't know why it didn't work now. Now it works. So this is basically your thickness, and what you can do is apply a lookup table, doesn't really matter, and then it will give you different colors for different thicknesses, if that makes sense. So that's very quick to do. Okay. And again, you could quantify it in 3D, but I didn't because of time.
Okay, perfect. Thank you.
Any questions on that? Okay, does anyone have specific questions for their own data, for their own analyses, where they think they can now have a discussion with us, pick a brain, I don't know, think about something?
I think the next step is to sort of think about the tutorial and see which steps we can apply to our pipelines. And I think unless I apply, I don't know what problems I'm going to face, right? And once you start doing them, then because you have run this data and you know this data in and out, so we ask you questions, we get stuck, we can solve them. I think it's how to modify the macro or how to apply it and what kind of parameters would you want to change depending on how large your blood vessels are, or if you're using this on neurons or any other data set.
No, I agree. And I think what I learned most was really to spend a lot of time just to look at the data and actually, I'll just stop sharing as well, and just really spend time on this enhancement and segmentation and what really improves your quality. Because again, every microscope is different, every transgenic is different, every vascular bed is different, and especially if you sometimes work with live data and then sometimes you work with fixed tissue, there are massive differences between them, and also the image quality. And it can be very challenging to find something that is kind of a box standard approach. So I'm not expecting that, you know, you will use my macro for all your data. I don't expect that, but that's kind of also why I thought today I use it and give you kind of a very broad overview and kind of give you starter points of what can we done, what is important to look at. And for example, like this 3D structure of voxels is something that I've learned the very hard way because I didn't know for about half a year in my PhD, and then I realized, and then, you know, so things like that are really, once you know them, it's fine, but if you don't know them, they can really make your life very, very hard. Same with like the contrast to noise ratio. It's like one of these reviewer comments of like, how do you know it makes your data better? It's like a standard, you need to show data instead of just saying it looks pretty or it looks better. It just, unfortunately, that doesn't work very well. Okay. Okay, it's 9:20 now. So we're actually quite early with kind of finishing. Are there any, so in my time, sorry, 9:20, I don't know why it's your time. I don't know if people are just fed up now, had enough, want a break, or want to hang around, have a chat. I'm more than happy to stay on. If you want to log off, you're more than welcome as well. I thank all of you really for joining in. Any feedback would be really more than appreciated. I will probably send you an email later this week as well, just kind of seeing how all of you get on and kind of what you took away from it, and how you take these 3.5 kind of hours to learn more, like really apply to your own data. And like I said,
when these videos will be online, so that if there are pieces we missed and we're like, wait, how did she get there?
I didn't even know how we would record until like a few hours ago, and then I was like, oh, are we? You know, so I have no idea. I'm very sorry, but we can find out. I can ask the organizers how and when, but I would assume that they just send a link afterwards to all of us. I would assume so.
Okay.
Access videos. Yeah, I will ask them. Right.
So I have a question. Do you ever get broken vessels, basically an area where maybe due to some problems with CNR, you don't get a continuous vessel? And is there a way to amend that, or do you just have to manually go and deal with those situations?
So very good question. This is my reviewer too at the moment. This is one of the things where we kind of, again, we don't live in a perfect world and data are not perfect. For us, what we see is that especially if you have highly pigmented fish, this can be an issue when you look at more dorsal vessels. However, we are mainly interested into the midbrain vessels where there's very continuous signal. And so for kind of more kind of broader insight, we're looking at about 150 odd vessels in the brains, individual vessels, and out of these it's about 3 to 4 which are normally broken. And so this is something where we say we can kind of live with it, because in the grand scheme of things it doesn't have this significant impact. It's because again we have this global quantification, we don't look at individual vessels at the moment. We hopefully will do in the future, but at the moment we look at all of them rather than just specific certain vessels. In terms of the question of like how can you amend that, that's something that I am looking into, but it's unfortunately quite challenging because so you could use like TensorFlow, automatic kind of fillings, etc., but it is quite challenging, and that's also why we started to look into this machine learning approaches. So that's the bioarchive paper that I've just shown you before, where you look into maybe if we use machine learning, we could just avoid this breaking of vessels, but also obviously working with data, we can improve the acquisition. So for example, you can improve it with using fish which have no pigmentation or have tissue clearing or have additional signals. So that's also one thing that a lot of people do. They use 2 different markers and then just overlap them, and this overlap just increases the quality. And so these are steps that you can take. So there are various different stages. And I think it's always kind of the question of how perfect do you want your data to be, kind of thing. So at the moment it's implemented for kind of the normal data. It's not these perfect, you know, highly optimized data. But obviously if you start with really good data and a high signal, then you can apply to or you will have less of the breaks, etc. If that makes sense. Octavia, thank you for joining us. Sorry that you have to leave. I hope you learned a lot by Octavia. Okay. Any other questions? No? Okay. So then again, if someone wants to hang around, I will be here for a bit longer. We can chat about individual data. If people, like I said, had enough, feel free to leave now. I thank you again for joining. It was really a pleasure to meet all of you. And I hope again that it was useful to all of you and that you learned something for your own data.
Yeah, thanks a lot.
Thank you so much.
Thank you.
Thank you. Thanks. Bye.
Nice to meet you.
Thanks.
It was fantastic. Thank you so much.
Thank you. Bye.